Sumita, good morning. Sumita, good morning.
গুড মর্নিং শুনতে পাচ্ছ?
ভালো? হ্যাঁ, শুনতে পাচ্ছি। ভালো আছেন তো?
হ্যাঁ, একদম একদম।
হুম।
তোমার লেকচার আছে?
আমার লেকচার আছে 11:30 থেকে। नमस्ते चौगले साहब, नमस्ते।
अरे
नमस्ते सर, गुड मॉर्निंग सर।
आपस में बात कर रहे हैं। हेलो, वेरी गुड मॉर्निंग सर।
गुड मॉर्निंग।
Well, a little bit late. Just right after is coming.
नमस्ते। केसरी साहब, नमस्ते केसरी साहब, गुड मॉर्निंग केसरी साहब।
नमस्ते, गुड मॉर्निंग, गुड मॉर्निंग।
Good morning, sir.
गुड मॉर्निंग।
आलं मीच माझ्या चष्म्यावर बसलो.
all of my
हमारे मुख्य अतिथि हम लोग आ गए हैं मंच पर। सबसे आग्रह है कि अपना अपना सीट पर बैठ जाएं। रतन बाबू हमारे विभाग के अध्यक्ष महोदय, कृपया अपना स्थान ग्रहण करें। ई सेमिनार कर रहा है, जिसमें कि लगभग हाइब्रिड मोड में कुछ लोग यहाँ फिजिकली मौजूद हैं और हमारे मुख्य रिसोर्स पर्सन ऑनलाइन डिफरेंट डिफरेंट कंट्री से जुड़े हुए हैं, डिफरेंट डिफरेंट शहरों से जुड़े हुए हैं, इसी देश के बहुत छात्र जुड़े हुए हैं। और ये लाइव टेलीकास्ट यूट्यूब लाइव पर भी चल रहा है। इसके अलावा गूगल मीट से सारे डेलीगेट्स एंड रिसोर्स पर्सन अभी ऑनलाइन है और ये सब हमारे साथ समक्ष हैं। आज के इस उद्घाटन समारोह में कुलपति चंद्र प्रताप सिंह, माननीय प्रति कुलपति महोदया प्रोफेसर डॉली सिन्हा, हमारे डीन फैकल्टी ऑफ साइंस प्रोफेसर रतन कुमार चौधरी और इस विभाग का अध्यक्ष हमारे प्रोफेसर कुमार मंचासीन है। कार्यक्रम को बिना डिले किए हुए, क्योंकि ऑनलाइन है, टाइट प्रोग्राम है मेरा, शाम को 5 बजे तक लगातार टेक्निकल सेशन है, मैं समय नहीं बिल्कुल लेना चाहता हूँ। सर्वप्रथम मैं माननीय कुलपति महोदय को पुष्प से उनका स्वागत कर रहे हैं और उस पुष्प से स्वागत के लिए हमारे विभाग अध्यक्ष प्रोफेसर कुमार उनको पुष्प देकर करेंगे। प्रिंसिपल साहब प्रेम कुमार प्रसाद जी स्वागत करता हूँ, जो आज के इस कार्यक्रम में जो अलगी पर कन्फाइंड है, उसके लिए वन ऑफ द फाइकोलॉजिस्ट ऑफ दिस सिटी, प्रिंसिपल सीएम साइंस कॉलेज प्रोफेसर प्रेम कुमार प्रसाद जी से आग्रह है कि हमारे माननीय प्रति कुलपति महोदया को उस क्वेश्चन देकर उनको स्वागत करें। आपसे आग्रह है कि आप प्रोफेसर रतन कुमार चौधरी जी को कुछ देकर स्वागत करें। उससे सम्मानित करना चाहता हूँ। कार्यक्रम को शुरू करने के लिए मैं वेलकम स्पीच और सब्जेक्ट के थीम को बताने के पहले कार्यक्रम का हम लाइटिंग ऑफ लैम्प के द्वारा शुरुआत करना चाहते हैं। अतः सभी अतिथिगण से अनुरोध है कि जो डैश पर मौजूद हैं वो कृपया दीप ज्वलित करके इस कार्यक्रम का शुभारंभ करें। उद्घाटन होते ही मैं सर्वप्रथम इस विभाग के विभागाध्यक्ष प्रोफेसर अभय कुमार जी से आग्रह करता हूं कि वो आकर माइक पर हमारे गेस्ट को स्वागत करें और उनसे यह भी आग्रह करता हूं कि आप विषय प्रवेश सारे जो यहाँ मौजूद हैं या ऑनलाइन लाइव यूट्यूब पर हैं उनको सब विषय प्रवेश करके स्वागत करें। प्रोफेसर अभय कुमार। Principals of different colleges and teachers, students, research scholars, media persons, and all the persons who are present here online and offline both. Firstly, I would like to honor our Honorable Vice Chancellor, Professor S.P. Singh, one of the visionary Vice Chancellor whose vision both in the structural and the functional aspect of the university is fascinating. Whether it is the construction of the new building or the renovation of the old building, beautification of the campus, regularization of the session, upliftment of the academic activities both in the teaching and research, in all aspects, you have the roadmap, and with this roadmap, undoubtedly this university will get an excellence in recent times. So we are proud of having a vice chancellor like you, and under the guidance, under the good wishes, I think this seminar will be successful. I wish to extend my warm welcome to Honorable Pro Vice Chancellor Professor Gauri Sinha, who is also well devoted to upliftment of the academic activities, academic standard of this university. Recently, we have seen the conference and workshop of plagiarism, where I think in the near future, this plagiarism will make us to produce more original research, more original PhD thesis in the university. From the days of announcement of this seminar, and when we got a meeting with Honorable Pro Vice Chancellor, a constant encouragement and guidance of regarding this seminar we got from herself, and that is the result that we have organized this seminar, e-seminar, and I think this will be successful with her best wishes also. I also extend my welcome to Dean Professor Ratan Kumar Chaudhary for his support and encouragement during organizing this seminar. I also welcome the principals who were the teachers of the botany earlier, and they gave several valuable suggestions in organizing seminar. I extend my welcome to head of departments of different faculty, teachers of different subjects who are here, and research scholars, students of PG, and other persons who are present here, and also the students and research scholars who are online. I also welcome the resource person who are online, though they are not here but well connected to us online. And I extend my heartiest thanks to them for sharing their time and valuable lecture. Our resource persons there are four resource persons for this seminar. One of the resource person, Dr. Bhavnath Jha, and FNA, who are the student of this university, who are the teacher of this university, and ultimately the Department of Biotechnology was opened by him. So he is the one of our resource person. He has worked on the algae seaweeds of the East Coast. Earlier, he was the director, Center Marine Research Institute, Bhavnagar. He is now retired. So, one of the resource person is Dr. Bhavnath Jha. The second resource person is Professor Samit Rai. He is the professor, Department of Botany, Vishwabharati University, Shantiniketan. He has written several books and published so many papers, and his areas of research is cytomorphology, cytotaxonomy, and cyanobacteria. Another resource person, Professor V.V. Chaubale, is an emeritus professor and at present he is associated with Bharati Vidyapith Pune, and he is working as a consultant on biodiesel from marine microalgae in collaboration with the Reliance industry. He has a very big project of biodiesel from this microalgae. Also, a member of the review committee on mission mode program on seaweed cultivation and utilization. Our fourth speaker, Professor J.P. Kesri, is Professor Center of Advanced Studies in Botany, University of Burdwan, West Bengal, and he is specialized in taxonomy and ecology of the fresh water and marine algae. He has also published several books, monographs on algae. So, all the resource persons are distinguished phycologists, distinguished algologists of India, and we are very thankful to them who have given time for their lecture. Now, coming to the seminar, the topic of the seminar is algae and human welfare. We all know that the 70% of the Earth's surface is covered with water. And second point is that the population of the world is fastly increasing. So the people have started using the agricultural land for their habitation. So there is no production of food matter. We have the plenty of food matter, but in spite of that, we have to turn our attention to the water body, and where algae is the habitat of that very particular aquatic system. As we have algae, we have the reference in the epic of the Kalidas अभिज्ञान शकुंतलम, और जिसमें उन्होंने लिखा है कि ये जो अलगी जिसे हम शैवाल या काई भी कहते हैं, तो वो शैवाल कमल के चारों ओर इस प्रकार लिपटा हुआ ग्रीन ग्रीन शैवाल चारों ओर लिपटा रहता है, जैसे किसी नारी के चारों नेकलेस गर्दन में नेकलेस का अपीयरेंस देता है, having the beauty of the lady. So we find the reference of this algae in the epic of the Kalidas. If you go through the algae, it is of different colors. It is green algae, red algae, blue-green algae, yellow algae, and different colors of algae. So we have plenty beautiful colors of the algae in nature, and some algae are in fresh water, some algae are of marine habitat. If you go through the biochemical aspect of the algae, you will find that algae are very rich in protein, carbohydrate, vitamins A, B, C, even formic acids. We find iodine in algae also. As regards the protein, some of the algae have more than means in soybean there is 28% protein, but in algae we find more than 50% protein. Even in one alga, that is Westellopsis, on which I have done work, and I found about 58% protein. One of the alga, that is Spirulina, and it is so rich in protein and so nutritious. The market, even our Ramdev also sold the dried powder of this alga in seed packets. So it is used as a food supplement. So algae is helping in our food supplements, where in India it is very important because we find malnutrition in spite of our economy is high. In spite of that, there is very malnutrition. So if we algae as a food supplement, naturally the malnutrition will be overcome. Things that we get from this algae are agar-agar, carrageen, etc. Right from early in the morning to the late night, we use different types of creams. We use different types of ointment or anything which is squeezed from a tube. That is due to the byproduct of this algae, that is agar-agar, carrageen, alginic acid, etc. The seaweeds have rich content of iodine, and one of the diseases due to the deficiency of iodine is water, and we can overcome by using this algae. In some of the countries like Japan, China, etc., algae are used as food water. Algae are also used as feed for cattle. One of the main use of the algae is also that it fastly reproduce, and in space people carry one of the algae, that is chlorella, and it multiplies so rapidly that deficiency of oxygen is overcome in space. So we find algae having many uses, have such an economic importance. Algae that is playing a very important role in human welfare. And one of our resource person, Professor J.P. Kesri, had the topic algae in glamour. Even our cream, our lipstick, our etc. are made from the product of the algae. So, keeping all this usefulness of the algae, of this seminar has been organized to make awareness about the algae, because this Mithilanchal is full of ponds where algae are found, so many algae are found. So we have to explore the algae, their byproduct, their products, and how it will be help to human beings. So I think with the lecture of our resource person, the seminar will be successful and an outcome will come. Thank you very much.
Thank you very much, Professor Algae Kumar, HOD of this department, has given a bright and clear-cut idea about how the algae is affecting human life. And in future too,
algae is going to be one of the important parts of the human being. Algae is a plant, but without leaf, without stem, without root, and without flower. Algae is a group of plant that doesn't have these things, but the all good quality of plant is having in a very confined way, in a very original way, in a very minute way. And the efficiency of the algae is far more than the local or land habiting plant. Hence, this minute microscopic algae, which is basically hydrophytic in nature, is a lot of capability to change our future world. And the aim and objective of this e-seminar is confined to the algae for human welfare. How this algae is going to be affected human welfare. In this context, I request Professor Algae Chaudhary, you please come on the gas and say a few words and good wishes for this seminar. Professor Algae Chaudhary, Science Chemistry.
Thank you. Thank you, Professor Sahu. Honorable Vice Chancellor, Professor S.P. Singh, Pro Vice Chancellor of the University, Professor Dolly Sinha, Madam, the Head of the Department, Professor Abhay Kumar, sitting on dais. Core principal belongs to botany, sitting in front. Principal CM Science College, Principal MRN College, Principal Miller College, and Principal ML College. Our former head and dean, Professor Sheila, Head of the Department of Zoology, Professor B.S. Jha, Head of the Department of Physics, Professor Mark Singh, faculty members of this department, teachers coming from various colleges belongs to botany, who is attending is participating this e-conference online, delegates, students, research scholars, the proctor of the university, Professor Ajay Jha, and media persons. I would like to thank our Honorable Vice Chancellor and Pro-Vice Chancellor, by whom initiative this of e-conference is started today from Botany Department. Of e-conference of every department, one of national level and another of international level. After the joining of these, in the meeting of heads and deans, they given a task that one national and one international conference be organized by each and every postgraduate department. And I thank the of botany because the first e-conference, national e-conference of this series is starting today. So I'm giving this thank. I'm a student of chemistry and algae, the subject algae for human welfare. So without chemistry, there is no existence of plants. So in each and every plant, the inside is chemistry. Since the prehistoric times, several seaweeds have been used as direct source of food. Algae is also one of them. We have almost various varieties of algae, various types of algae. Algae has protein, vitamins, minerals, and so many chemicals having different values present in the algae. So it is very helpful in the human welfare. Not only as the food or food supplement, algae is also used to eradicate the pollution. It absorbs carbon dioxide, and by absorbing the carbon dioxide, it provides pure oxygen for the human being. Most important part of diet as algae was taken in Japan and China. And in these days, also in South India, the algae is a good source of food for the South Indian people. People have used algae as food. It also used as fertilizers for 100 years. More recent use of algae include biodiesel fuel, thickening agent of food, bacterial growth medium, and pollution control. Algae is fit to grow 10 times more rapidly than the terrestrial plants. It helps in controlling pollution. Instead of increasing the amount of toxic chemicals to aid in the removal of sludge and poisonous organic molecules from our water resources, they use algae. 30,000 species of algae are reported, as I came to know. A wide range of biochemicals are produced by these species. They are used in medicine, food, nutrients, dyes, cosmetics, oils, biofuels, biomethane, bioethanol, vitamins, minerals, antioxidants, pigments, and beta-carotene, etc. These all chemicals produced by algae are non-hazardous and frequently used in environmentally eco-friendly green chemistry. So, we have learned that every slope in India is a mantra, and every plant of India is a medicine. Every slope is a mantra, and every plant is a medicine. So, I once again thank the Department of Botany for choosing such type of popular topic, algae for and human welfare. So it is very commendable, and thanks the Department of Botany who gave me opportunity to come here and dash. Thank you very much.
बहुत बहुत धन्यवाद सर, आपने दो बातें जो अलगी के बारे में जो लोगों को बताया, अपने आप में बहुत ही महत्वपूर्ण था। नेक्स्ट स्पीकर के रूप में, आशीर्वचन के रूप में मैं प्रोफेसर डॉली सिन्हा जी को बुलाना चाह रहा हूँ कि वो आए यहाँ पर मुझसे बोले। लेकिन मैं के बारे में कुछ बताना चाह रहा हूँ। ये जो सेमिनार होता है, इसमें बेसिकली ऑब्जेक्टिव तो होता है टेक्निकल चीजों को लोग के पास रखना, रिसर्च के संबंध में होना, लेकिन इसके साथ कई तरह के चीज इनलिंक हो जाते हैं। जैसे अभी माननीय प्रतिकुल मंत्री महोदय के वह विजन था कि आप हर एक डिपार्टमेंट में इंटरनेट लगाइए, जो इंटरनेट के कारण नहीं चलता है। हमने अभी के लिए हाई स्पीड इंटरनेट जो यूट्यूब लाइव चल रहा है, सब लोग देख रहे हैं, रिकॉर्डिंग हो रहा है ऑनलाइन क्लाउड पे, और ये इनका पूरा दिशा निर्देश इन्हीं का था। और ये दिशा निर्देश से हमने ये टेक्नोलॉजी को हमने इस विभाग में स्ट्रीमलाइन कर दिया है, हैसल फ्री कर दिया है। फ्यूचर में ऐसा व्यवस्था कभी हम कभी सोच भी नहीं सकते कि व्यवस्था आ जाएगी। इनके पूरा श्रेय हमारे माननीय कुलपति महोदय जाता है। और इसके विभाग में ये हाई स्किल इंटेंट आने के कारण हम लोग आगे भी जो माननीय कुलपति महोदय ने पहले कहा था कि इनोवेटिव कुछ आइडिया कीजिए, हमने एक प्रोजेक्ट डाला है जिसमें कि इस टेक्नोलॉजी का पूरा इस्तेमाल हो जाएगा। अब मैं आग्रह कर रहा हूँ प्रोफेसर डॉली से अपना ये आशीर्वाद से हमको दीजिए जो कि यह सेमिनार अच्छा से कर सके। माननीय कुलपति महोदय डॉली सिन्हा।
छोटा बड़ा होता जाएगा,
निकल जाता है हाथ से।
कोई बात नहीं, आपकी
Namaskar and very good morning to all present here. We have on the dais distinguished personalities, Professor S.P. Chancellor, Ellen Mitchell University, Dr. Algae Kumar, Head, University Department of Economy, Dr. Ratan Chaudhary Sir, Faculty of Science, and we have equally distinguished personalities in the audience, the university, as well as on my own. I welcome you all once again in this seminar. On behalf of the university and my own, I also welcome the distinguished speakers of today, Dr. Bernard Jha, Dr. Samik Ray, Dr. B.B. Chogule, Dr. J.P. Kesri. We are indeed very grateful to the for accepting our request to enrich our faculty members and research scholars today. The credit of organizing this seminar goes to the teachers, research scholars, and students of the department. I congratulate all the members of the organizing team that you have chosen a very fascinating topic and human welfare. As we have listened our previous speakers who had already explored and said many important things about algae. Biologists explore nature and in turn find out natural materials which are of use to human for food and other needs. To fulfill the demand of global population, there is a constant search for new food sources. We get about 90% of our food from land. But in India, we are very fortunate that we have a rich diversity of higher plants from which we get many useful products in plenty. We therefore have not given much attention to algae, which is a fascinating group of aquatic organisms and quite rich in protein, vitamin and mineral, including iodine, which is very essential for the welfare of human beings. The possibility of its use as food and other use was also not explored very seriously in India till recently. As our previous speaker has mentioned, that recently only in South India we have started using West Bengal also it is used for fodder for domestic animals. So in this context, I once again congratulate the department for choosing this topic which is very relevant, and I also expect that our research scholars and students will be highly benefited as the distinguished speakers will explore the fascinating world of algae. There are about 7 types of algae, each having thousands of species. So there is a rich scope for research in this area. And as we know that the new education policy emphasizes on research even in undergraduate level, so this could be a very good field of research and our students can do experiments in this area and come out with some success. And we know that nutrition is an area which have lots of water bodies and growing algae will not be a problem. So this is what I would expect from the department and from the head of the department, I would expect that message will be given to the various colleges where the teaching of undergraduate or postgraduates are going on, they can also research. And if we can do something which is cost effective, this would solve many problems. We know that Mithilanchal is also known for its domestic animal and milk produce, and it can be a very rich source of food for our domestic animals. So I have great expectations. This is just the beginning. The botany department has started and the path to others. So once again, I congratulate all involved in organizing this seminar. Thank you, all of us.
धन्यवाद मैम, आपके आशीर्वादों से सेमिनार अच्छा होगा, हमें पूरा विश्वास है। हमारे माननीय कुलपति महोदय बहुत विजनरी है, बहुत ही इफेक्टिव है, बहुत ही अनुभव है, बहुत ही हृदय बड़ा है, इसमें कोई दो मत नहीं है। माननीय कुलपति का विजन इफेक्टिव है, इसका कुछ ही कई लेवल के काम दिखा पड़े। माननीय कुलपति ने जो रिसर्च के एक्सेलेंस करने के कई कदम उठाए हैं, जिसमें जैसा आपको मालूम होगा कि कल हम लोग क्या था प्लेगरिज्म के बारे में, जिनका पूरा विजन था रिसर्च की क्वालिटी तभी इंक्रीज होगी जब आप क्वालिटी वर्क करेंगे, वर्क करेंगे। किसी का पहले से लिखा लिखाया पैटर्न पर जब काम करेंगे तो वो रिसर्च में नहीं आता है। और यूनिवर्सिटी का वन ऑफ द ऑब्जेक्टिव या रिसर्च ही है, और रिसर्च में एक्सेलेंस स्टेट लेवल का नहीं, नेशनल लेवल का नहीं, इंटरनेशनल लेवल का हो, ऐसा हम लोग रास्ता खोजें। सर का बहुत अच्छा डायलॉग होता है कि जब कभी भी हम प्लान करते हैं तो शॉर्ट टर्म प्लान होता है, लॉन्ग टर्म प्लान होता है, और एक बहुत बड़ा प्लान होता है, पर्सपेक्टिव प्लान होता है। सर का सारा काम पर्सपेक्टिव प्लान से होता है, एक लॉन्ग टर्म विजन लेके चलते हैं, जिसका शुरुआत आते ही 2 महीने में उनकी नींव इन्होंने डाली है। नींव के तहत हमको ग्रो करना है। हमारे लिए ऐसे महान माननीय कुलपति लीडर हमारे हैं, हमारे दर्द की बात है। आज के दिन जो इस सेमिनार आयोजन हो रहा है, उनका पूरा विजन सर का था और उन्होंने कहा था कि शुरुआत करें। हम लकी हैं बृहस्पति विभाग, लकी हैं के साहू कि इनके विजन को तुरंत हम पकड़ते हैं। इनके 3 प्रोजेक्ट में सबसे पहले बृहस्पति विभाग आके काम किया है, जिसमें एक ई सेमिनार है, प्लेगरिज्म है और इनोवेटिव प्रोजेक्ट जो स्टूडेंट सेंट्रिक है, बायोडायवर्सिटी ऑफ एलन मिथिलांचल एक डिजिटल कंसेप्ट प्रोफाइल बनाने का, जिसमें भी पैसा नहीं लगता है, के द्वारा होगा। ये तीनों का पूरा नींव डालने की क्रिया हमारे माननीय कुलपति के दिशा निर्देश हुआ और हमने दिखाया, इसमें हमको थोड़ा गर्व होता है। मैं बगैर समय लिए हुए माननीय कुलपति महोदय से आग्रह करता हूँ मेरे आप अपना आशीर्वचन दे और मुझे ऐसे गुण और बताएगा आगे मैं अच्छा पढ़ सकता। माननीय कुलपति।
of the Algae University, the person of the academics, and the person of the think for the welfare of everybody, even the university dean of the science, Mr. Balaji, head of the botany department, Professor Abhay, head of the geology department, Professor B.S. Raji, and the sports officer of the university, Professor Ajay. Organizer of this wonderful seminar, Professor Sam. Principals of the different constituent colleges, faculty members, dear students, Mrs. Brothers. Professor Sheila, retired from the department. It's the wonderful occasion for the LNB for the department itself. What I think. It's a wonderful occasion for the that the university is moving ahead. Accordingly, the design path. So everybody is moving, that's the one point. But the designated towards the movement towards the designated direction, objectives, goals, and moving with the objects. It's a different task. Speed, it's a different task, and moving towards target and towards the goal. And that is the perspective goal of the university entire. It's a different task. Really, I congratulate the departments to moving ahead with the definite targets or the definite goals to achievements of the excellence in the university itself. Difficult task for the any university, for the any individual to compare the to others or not to compare to others. It's a dilemma. If you are not going to compare yourself with the others and compare on the basis of the thoughts you are developing without the action, I think it's not the definite path. If you are going to compare yourself with the action which has been taken by you to achieve the goals and other things, what you have decided earlier is a different task. And it's a fruitful task. Exercise, everybody is doing exercise, doing things. But towards the definite goal, achievement is always measured towards the journey towards the goal. So ultimate goal is defined by the ultimate authority of the higher education. Excellence in the higher education. Quality in the higher education. Definitely coming to the university system to just take up the class. Sufficient knowledge. Going the class regularly on the time and come out after taking the class. So one way to impart in the higher education, way the out of box to compare to the other, the comparison to the other. It's a difficult task, it's a real task. With the national education policy, the other policy, what has been decided by the apex body of the higher education system, it's a different. Although apart from that, leaving all these things, it has been designated by the other speakers. Designated designs for ourselves. It's a big question. One that is related with it's a very important task. That being a human, being an individual, you would like to wish to achieve something. And one more important thing is that achieve the happiness in one time. The idea of the everybody, the target of the everybody, target of the human beings, with the brain, with the rational behavior, is to enjoy the pleasure. Whether this pleasure, whether this enjoyment is going to affect the only individual, to affect the majority of the people, or the human being itself. So science has the balance. Always have science things and do the things and prefer to do the in larger interest of the people for the human being itself. Whether in field of the medicine, as you said, that algae is in the field of the medicine, it's useful for that. Field of the medicine, fields of the glamour, field of energy, food. It's searching for a food of the human being at large, not for any individual. And it's the beauty of the science. It's not the beauty of the social science. It's the beauty of the science that science always thinks about the large perspective, larger perspective. Because the goal of the science is if you desire that you do the thing for the interest of the only individual, you can't do it. Because the outcomes of the science is come through the technology. And technology is for everybody. Technology is not limited for limited man. I'm a student of the economics in that way. Types of the goods in the nature. One goods is known as the individual goods. Another goods is known as the public goods. So public goods means without paying a cost, you have authority to enjoy that fruit of the public goods. One thing. And the second merit of the public goods is that if you are going to utilize the public goods in favor of your usage, the share of the other people is not going to reduce. It's the beauty of the public goods. It's not going to share of the reduce for anybody else. And what is the concept of the welfare? That is only the concept of the welfare. If you are using the consuming anything as a consumable item, the share of the other people is not going to reduce that. Like education. If some students are getting more marks in the class, in the system, it's not going to reduce the percentage of the other people. It's going to extend the percentage of their own itself. So I think the topic, what has been chosen in the terms of the human welfare, it has a great perspective. Not only in terms of the science and technology, not only in terms of your specific subject, but in the greater interest of the people itself, the greater interest of the universe. And that aspect always has been the search of the science, human aspect. Another aspect of the subject of the education system. And to create, to observe, to sustain, to extend this larger interest of the human welfare is the objective of the higher education, it's the objective of the education itself. So in that way, the department, the organizers, Sahu and other teams of the departments for choosing this that is related with the human welfare, going to the fruits for the human welfare, producing to the fruits for the human welfare. As per the students, the researchers, what has been told by the Vice Chancellor and other people, if you told anybody, whether expert people are there, the national level, the field of research, the area of the discussion, they will be greater, I hope, in terms of this national seminar, we get for the system, providing this opportunity for this, in this LL Mithila University to head towards the goal. Really
I congratulate to all of you for that. This achievement, greater achievement, and greater what has been done by you. Thanks.
Thank you very much, sir. We are certainly in need of going towards the welfare of women in all perspective level. किसी भी सेमिनार का अच्छा होना कुछ बेसिक बातों पर उभरता है। इसमें कितने बच्चे हैं, कितने रिसर्च स्कॉलर्स आए, कितने रिसर्च पेपर आए, कितने अच्छे पर्सन अवेलेबल, बहुत कम समय में 10-15 दिन के अंतराल में मुझे आज गर्व है इस बात का कि मेरे पास हाई क्वालिटी 26 एब्स्ट्रैक्ट आए हैं पर्टिकुलर दिस टॉपिक पे। यह ब्रॉड टॉपिक नहीं है, बॉटनी या लाइफ साइंसेज नहीं है, यह बॉटनी का एक खास पार्ट, बहुत माइक्रो लेवल का एक पार्ट है। उस अलगी पर इतने पेपर आ जाना कम समय में, यह भी एक अच्छे इस सेमिनार का एक द्योतक है। साथ में जिस फील्ड के हम काम कर रहे हैं, जिसको फाइटोलॉजी कहते हैं, एल्गोलॉजी भी नाम है इस फील्ड का, इस फील्ड के टॉप मोस्ट व्यक्ति इंडिया के हमारे छोटे से आग्रह पर अवेलेबल हो गए, यह भी एक गर्व की बात है। हम जानते हैं कि ये आज के जो सेशन स्टूडेंट का होगा, जो पेपर पढ़े जाएंगे और रिसोर्स पर्सन जो अपना हाई लेवल के क्वालिटी बात को कहेंगे, इस सब का संग्रह आने वाले समय में मुझे उम्मीद है कि एक बुक में संग्रह होकर प्रकाशित की जाएगी। हमारा आज का जो कार्यक्रम है कई सेशन में बटे हुए हैं, कई रिसोर्स पर्सन का प्रेजेंटेशन है कई तरह से, लेकिन अभी का यह इनॉग्रेशन सेशन जो हमारे समाप्त की अवस्था में है और इस दिशा में आगे करते हुए मैं इस इनॉग्रेशन सेशन के वोट ऑफ थैंक्स के लिए इसी विभाग के स्टूडेंट और टीचर रहे हुए जो अभी प्रिंसिपल है हमारे कॉलेज के, डॉक्टर अनिल कुमार झा जी से आग्रह करते हैं कि आप मंच पे आके वोट ऑफ थैंक्स दें।
अतिथि के रूप में उपस्थित ललित नारायण विश्वविद्यालय के माननीय कुलपति महोदय, माननीय कुलपति महोदया, आभासी रूप में इस सेमिनार से जुड़े हुए सभी सम्मानित संसाधन पुरुष, मंचासीन अध्यक्ष विज्ञान संकाय, विभागाध्यक्ष वनस्पति विज्ञान विभाग, कार्यक्रम का संचालन कर रहे श्री के साहू जी, सभागार में बैठे हुए विभिन्न विभागों के विभागाध्यक्ष महोदय, सभी उपस्थित सम्मानित शिक्षकगण, छात्र छात्राएं एवं मीडिया बंधु, वनस्पति विज्ञान विभाग द्वारा संचालित इस एक दिवसीय सेमिनार का उद्घाटन सत्र अब समाप्त की ओर अग्रसर है और मुझे विभाग द्वारा सभी सम्मानित अतिथियों के प्रति कृतज्ञता ज्ञापित करने की जवाबदेही दी गई है। निश्चित रूप से यह मेरे लिए गौरव का क्षण है। सर्वप्रथम मैं मुख्य अतिथि ललित नारायण मिथिला विश्वविद्यालय के माननीय कुलपति महोदय के प्रति आभार प्रकट करता हूँ, अपनी कृतज्ञता ज्ञापित करता हूँ, जिन्होंने अपना बहुमूल्य समय देकर अपने आशीर्वाद वचनों से हम सबों को अनुग्रहित किया है। महोदय, निश्चित रूप से आपका आगमन ही इस सेमिनार की सफलता का द्योतक है। हम सभी आपके आभारी हैं। हमें पूर्ण विश्वास है भविष्य में भी इस विभाग को आपका सहयोग, आपका आशीर्वाद इसी रूप में मिलता रहेगा और आपके कुशल नेतृत्व में यह विश्वविद्यालय नित नई ऊंचाइयों को स्पर्श करेगा। हमारे बीच माननीय कुलपति महोदया भी उपस्थित हैं। एकेडमिक एनवायरनमेंट को प्रमोट करने में इनकी महती भूमिका रही है और आपकी गरिमामयी उपस्थिति से यह विभाग आपका ऋण है। विभाग की ओर से हम आपके प्रति भी कृतज्ञता ज्ञापित करते हैं। हम आभार प्रकट करते हैं अपने सभी संसाधन पुरुषों का जिन्होंने तमाम व्यस्तताओं के बीच भी इतने महत्वपूर्ण विषय पर चर्चा के लिए अपना बहुमूल्य समय दिया है। हमारे बीच अध्यक्ष विज्ञान संकाय भी उपस्थित हैं। इनके एक्सपीरियंस और एक्सपर्टीज का लाभ विश्वविद्यालय को सदैव मिलता रहा है और हम सब आपके भी आभारी हैं। मैं आभार प्रकट करता हूं वनस्पति विज्ञान विभाग के विभागाध्यक्ष महोदय का, जिन्होंने इतने महत्वपूर्ण विषय पर इस सेमिनार का आयोजन किया और संबंधित विषय पर नई नई संभावनाओं से अवगत होने का हम सबों को उचित अवसर प्रदान किया। सभागार में बैठे हुए सभी विभागाध्यक्षों, सभी सम्मानित शिक्षकों, सभी छात्र छात्राओं के भी हम आभारी हैं, जिन्होंने धैर्यपूर्वक विभिन्न वक्ताओं के विचारों को सुना। हम उपस्थित मीडिया बंधुओं के भी आभारी हैं। बिना उनके सहयोग के किसी भी सेमिनार की सफलता की कामना नहीं की जा सकती। और अंत में इस कार्यक्रम का संचालन कर रहे श्री के साहू जी के प्रति भी हम आभार प्रकट करते हैं, जिन्होंने इतनी सफलतापूर्वक इस कार्यक्रम का संचालन किया। और अंत में आप सबों का पुनः आभार प्रकट करते हुए कुलपति महोदय की अनुमति से इस उद्घाटन सत्र की समाप्ति की घोषणा की जाती है। आप सबों को पुनः बहुत बहुत धन्यवाद। Hello. Professor Kesri.
Yes, hello.
हाँ आ गए आ गए आ गए आ गए उनको सेंटर में कीजिए ना हाँ
Hello.
Hello.
Kesri.
Yes, yes.
या
Are you listening me?
Yes, yes, I can listen. I can listen. I can listen. I don't know whether I am audible to you or not.
की आवाज नहीं आ रही है
आ जाएगा.
Yes, sir. Now you are audible.
Yes, I am audible.
Just.
Am I audible? Hmm. Hello. Hello.
सर बोलिए ना
Hello.
Sir, please start.
Hello. Can you listen?
Yes, yes, sir, continue.
Okay. सुनाई पड़ रहा है आपको? Okay. Professor Samit Rai is there. Professor Samit Rai, he is also an expert of charophytes, and I never met Professor Bhavnath Jha, but I have seen his book on civets of Indian civets, a book on civets, and I heard about him. So, to all these respected persons, head of the department, and all my colleagues present there, on my behalf and on the behalf of my university, I heartily acknowledge you this kind invitation. Now, before going into this lecture, I hope I present it. I just present it. Yes, sir. So, is it the screen on the screen? Hello? Hello?
Yes.
My topic is on the screen.
टॉपिक कर रहे हैं स्क्रीन पर
स्क्रीन पर आ रहा है डॉक्टर साहब?
Yes, sir.
अच्छा, ओके, थैंक यू। तो मैं स्टार्ट करता हूँ। मैं थोड़ा सा चूंकि हिंदी मेरी मातृभाषा है, इसीलिए मैं थोड़ा सा मिला जुला के बोलूँगा। ये मेरे लिए भी अच्छा रहेगा कि हिंदी में बोलने का मुझे बहुत कम मौका मिलता है, तो आपके साथ मैं हिंदी में शेयर कर पाऊंगा, तो ये मेरे लिए बड़ी खुशी की बात होगी मेरी मातृभाषा में। सो धन्यवाद इस इनविटेशन के लिए। यहाँ मैं समझता हूँ कि कुछ लोग ऑनलाइन भी हैं और कुछ लोग यहाँ पर आपके ऑडियंस में भी होंगे। सो ये जो टॉपिक है, दिस इज एल्गी इन ग्लैमर, अगर कहीं कोई असुविधा हो तो मुझे थोड़ा सा इंटरप्ट कर देंगे, क्योंकि ऐसा है कि मैं ऑडियंस को यहाँ देख नहीं पा रहा हूँ कहाँ क्या है, क्योंकि ये फिर स्क्रीन पे फिर मेरा पूरा लेक्चर ही है। खैर उससे उससे कुछ फर्क नहीं पड़ता है, लेकिन मैं यहाँ केवल अपने टॉपिक को देख पा रहा हूँ, तो इसी के आधार पर मैं पहले इसको स्टार्ट करूँ। तो पहले इस लेक्चर का फर्स्ट जो है मैं यहाँ बताऊं कि पहले ये देखिए, ये एल्गिन ग्लैमर अभी चाहे पुरुष हो या स्त्री हो, दोनों अपने आप को सुंदर दिखाना चाहता है, और इसके लिए बहुत सी चीजें अभी आ गई हैं। तो कोई भी लेक्चर इस तरह एक सुंदर लेडी के इमेज से शुरू हो, ये तो अच्छा, बहुत लोगों को अच्छा लगेगा। तो खैर यहाँ जो पहला मेरा ये है, ये देखिए पहले स्लाइड में ये आप ये जो स्ट्रक्चर है, ये स्ट्रक्चर है, ये एक वुडन पेंडेंट है, ये काठ का बना हुआ एक पेंडेंट है। ये हमें प्रोफेसर, ये हमें ओके,
इसकी आवाज ज्यादा है।
हेलो प्रोफेसर यामागिशी, हेलो, आवाज ठीक कट जा रही है क्या आवाज? हेलो,
नहीं सर, क्लियर है सर।
अच्छा ठीक है, तो मैं लेक्चर में चालू रखता हूं। तो बात ऐसा है कि मेरे ये जो मैंने ये अभी एक वुडन स्ट्रक्चर आपको दिखाया, दिस इज एन वुडन पेंडेंट एंड इट इज एक्चुअली मेड बाय वन ऑफ द सीनियर फाइकोलॉजिस्ट्स ऑफ द वर्ल्ड। मैं समझता हूं कि ही इज मोस्ट एजेड फाइकोलॉजिस्ट इन दिस वर्ल्ड। He is Professor Takaaki Yamagishi. His date of birth is around September some day in 1923. So he is now running 96 plus 97, Professor Takaaki Yamagishi. Actually, this Professor Takaaki Yamagishi, he is a friend of my teacher, Professor Pranjit Sharma. My Professor Pranjit Sharma, he is 80 plus, but Yamagishi is 96 plus, 97 around. So Professor Yamagishi, I should say about Professor Yamagishi, he was a professor in Tokyo, in some Osaka university like that. From there, he retired in at the age of 65, and then he instituted a laboratory in his home premises, and as usual he start his day with the time that he used to go to university, and he used to sit in that laboratory. Now, while in his laboratory, this laboratory is known as Tokyo Phytoplankton Institute, and Professor Yamagishi is extremely famous for this phytoplanktons. He is a very known worker in the phytoplankton taxonomy and ecology like that. Now, he used to stay in his laboratory for long time, and in a day, and his laboratory is open to all phycologists of Japan. Everybody can go and sit in his laboratory. He can work, he can consult his library, and the coffee mug is always ready. So this is his routine. Now, when he get his spare time, जब उनको खाली समय मिलता है, कभी वो फाइटोप्लैंक्टन माइक्रोस्कोप पे अच्छा नहीं लगता है, तो क्या करते हैं कि ये वुडन क्राफ्ट वर्क करते हैं, वुडन क्राफ्ट वर्क करते हैं एंटरटेनमेंट के लिए। तो दिस वुडन क्राफ्ट वर्क he made as a pendant, and about 25 years or 30 years back, he has given me as a gift. This is about 2.3-inch diameter pendant of wooden craft exclusively made by him. And you can see that there is a excellent design on this pendant in this craft work. Now, this inspired me much. I find that this is a replica of one phytoplankton. This is the phytoplankton, real photograph taken in my laboratory. This is Pediastrum. It is a floating phytoplankton. Now you can see one electron microscopic image of this another, that same phytoplankton, electron microscopic, it is digitally colored. Then when I was preparing this lecture, I was searching in the net that whether these designs have been used, since designs are very excellent, marvelous, whether the designs are used for any purpose, making jewelry or making designer articles, etc. I find this is a platinum, a jewelry replica of Pediastrum. It is available in the net. If anybody can spare a huge amount of money, he will get this Pediastrum jewelry. Now this inspired me. From this inspiration, I prepared this lecture. Now this is one of the extended lecture of this. I have prepared in another form, Algae in Fashion and Design. So it will very big lecture. Therefore, I separated this lecture into two. One is Algae in Glamour and Algae in Fashion and Design. So this design, this is very interesting, that the algal fascinating designs, microscopic designs of, because phytoplanktons are extremely beautiful. If you see phytoplanktons under microscope, they are so beautiful that it will be impossible for you to get out of the microscope. So these are very beautiful. Now these beautiful designs, now they have been utilized in several designer things. Designer slippers are available, designer dresses are available, designer your coffee mugs are available, then dresses, then many other things, designer umbrellas are available, many things are available inspired by the design of algae. But I'm not going in that aspect. I am restricting myself with the glamour part. Now, what is glamour? Everybody know that to make yourself or exhibit yourself beautiful, you have to follow certain things. You should be internally, spiritually beautiful. You should be mentally beautiful. And last but not the least is physically beautiful. So if there are three things which, if you want to be beautiful or glamorous, then these three things should be linked. One is spiritual beauty, then your mental beauty, then your physical beauty. Now, physical beauty, how it is managed, in the most part of our country, or for male and female, you are using various type of cosmetics. These cosmetics are made of your herbal products or made of some petroleum products, etc. Many organic materials, inorganic materials, everything, your Fair and Lovely, everything. These are available, pond cream or aloe vera cream, everything which is available, that are applied to your skin. Now, the glamour, if you consider physically, then it has two components. If your physical health is not good, it will not be reflected in the skin. If you just simply apply the cosmetics on your skin, it will make your skin beautiful, it will make you beautiful, but it is not permanent. So in this lecture, I would like to concentrate on that part, how your skin or your body will feel better so that your skin will be beautiful. Now, you see, these are two real photographs of algae. One is Micrasterias radians and another is Micrasterias mahabulaesuarensis. These photographs have been taken by us in our laboratory and fortunately have been utilized in one of the felicitation volumes of my teacher. That book was edited by Professor Kargupta and myself, Glimpses of Indian Phycology. On the cover page, these two phytoplankton. You can see that this Micrasterias, in one of the precise I have seen that this Micrasterias, this beautiful microscopic algae, is considered as one of the most beautiful organisms on microbial earth, microbial flora, microbial world. So these radians, Micrasterias radians or mahabulaesuarensis, they have been utilized in making designer jewelry. Then I am coming to this is diatoms. These are actually the scanning electron microscope of diatoms. They are actually some algae made of, their cell wall is made of silica. They are made of silica. Well, I mean lecture achi ekhon kotha bolte parbo na. They are made of silica. They are made of silica. So this silica crustules, if you see this diatoms, you will find this electron microscopic images. They are so beautiful. These designs are extremely beautiful. Then diatoms, you can see the diatoms. These are the magnificent images of diatom crustules made of silica. These are the assorted images of diatoms. Then this is another, this is another, this is another, this is another, this is another. These are assorted images. This is one. And you see this another beautiful organism. These are coccolithophorids. You see that coccolithophorids, they are actually marine organisms and they have their cover having small scales. These small scales are made of calcium carbonate. Now, these coccolithophorids and probably the blackboard chalks, what we are using, they are made of this calcareous remains of coccolithophorids because their fossils they have been deposited in Cretaceous period and they are called land of Crete or Cretaceous period. But may not be all the chalk available with us are actually this coccolithophorids, but I have studied somewhere that many of the chalks, actually this blackboard chalks, may have been the source from this coccolithophorids. So they are very beautiful organisms. Now I was talking about these coccolithophorids, so these are actually used in the several designer jewelry. You can see these pendants. You can see here Micrasterias have been utilized here in making this pendant or diatoms for this making this pendant. These are the ear rings और ये अंगूठी है, ये भी algae से designs inspired है, ये सारे inspired by algae. Now this was about the designs or jewelry, but I am concentrating on the cosmetics parts. Now this biocosmetic term is extremely popular to many persons in the world, and people are actually fascinating with this because the chemical cosmetics, they can harm your skin or they may have side effects. So eco-friendly or nature-friendly biocosmetics are being used in this country also or all over the world. There is a laboratory known as Haas Laboratory. It is in Germany. Actually, I don't know why. This laboratory is actually preparing several biocosmetic products, and they are preparing all biocosmetics products from algae only. And I came to know that they have actually established a branch in Chandigarh. So we may go through it. So this, you can see, this is one of the cleansing milk. This is, you can see, it is fresh water, algae plus, and then aloe vera. This is cleansing milk. Then this is another Blue Lagoon body spa. You can see algae and minerals and body scrub. This is another. Then this is algae moistening kit. Actually, sometimes back, I have given a project in MSc to one of my students that to go and search whether your beauty parlors, they are using algal products or not. So when she was in one of the beauty parlor, they told that yes, we have algae moistening kit, but you have to order first because these are costly and regular customers are not using. Everybody who actually pay extra, then we can procure it. So these are the algae moistening kit available in the market for as a body caring cream. Now this is algae face mask. These are all available in the market. Then you see, this is one of very excellent examples. You see that female, they are using eyeliner, eyeshadow. These are the things. So these eyeliner and eyeshadow, you see these eyeliner, eyeshadow. These eyeliner or eyeshadow, this lady has used from a product of spirulina. Some of the senior professors in this university, he was talking about the spirulina. Now, spirulina, it is one of the fascinating alga having 60 to 70% protein, and it is blue-green alga having two pigments. One is the phycocyanin and one other is phycoerythrin. This phycocyanin actually is a blue pigment. Now, this blue pigment, he has used for his eyeliner, eyeshadow purpose, and the spirulina cosmetics are available in the market. In Amazon you will get, or whatever the other online options you will get them, but not in the regular markets. But I think spirulina or many other things are actually available. I have seen one of the your famous beautician Sahnaz Hussein's products, she has used some algal component also, and I have seen one soap, Dove, they have given this algal component in their cover. Now, this is another. मैं थोड़ा सा हिंदी में बोलता हूँ क्योंकि एक हम लोगों के यहाँ बंगाल में तो ये प्रथा लगभग चली गई है. एक्चुअली बिहार में, उत्तर प्रदेश में, इन सब जगहों में एक हम लोग शादी के समय एक प्रथा है लगाने की. 5 दिन तक जो लड़का और लड़की दोनों को उबटन लगाया जाता है देह में. उसमें हल्दी होते हैं और मसाले होते हैं. पूरे देह पर एक लेप लगाया जाता है. ये एक ट्रेडिशनल आप कह सकते हैं डिसइंफेक्टेंट की तरह है. तो इसका एक विकल्प जो अभी फ्रांस में एक थेरेपी आई हुई है जिसको बोलते हैं थैलासोथेरेपी. इस थैलासोथेरेपी में ये लोग क्या करते हैं, दे आर यूजिंग द मेरीन एल्गल पेस्ट और उसको ये बॉडी के ऊपर ये लोशन की तरह लगाते हैं. और ये लोशन के आधार पर होता क्या है कि इसको बॉडी में ये आर्थराइटिस में यार और ये स्किन के टोनिंग में, स्किन केयर में ये अभी यूज होता है. तो मैं एक बार सर्च कर रहा था तो मैंने देखा नेट पे कि दिस थैलासोथेरेपी is also available in some cities of India, particularly बॉम्बे और दिल्ली में हो सकता है. क्योंकि मेरे पास एक मेल आया था कि आपके लिए मैं बहुत सस्ते पे आपको थैलासोथेरेपी करवा दूंगा, $200 लगेगा. तो $200 मेरे पास था नहीं, मैंने थैलासोथेरेपी करवाया नहीं, लेकिन है अवेलेबल, इंडिया में भी है. तो इसके बाद हम लोग ये आते हैं कि यू सी, ये बहुतों का परिचित ये सीन होगा कि हिल स्टेशन पर you may have seen that this plants are bark is covered with stentifolia. Now this stentifolia is a good source of beta carotene. Now what is beta carotene? Beta carotene, actually a term, this term has came from carrot, and it is orange colored pigment. Hello? 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 This orange colored pigment, it is called beta carotene. Now this beta carotene is available in many algae. Now beta carotene has two roles. First, it is a precursor of vitamin A. Second, it is a very good antioxidant. Now, both the things I will explain one by one. First, beta carotene, this is the molecular structure of beta carotene, and this is the molecular structure of vitamin A, or it is called retinol. Now, if beta carotene, molecule of beta carotene is cut in the middle by oxidizing enzyme, you will get two molecules of retinol or vitamin A. That means when beta carotene enters in your body, it is oxidized into two molecules of vitamin A. That means beta carotene actually is the precursor of vitamin A. So we are also using beta carotene as a source of this beta carotene as a source of vitamin A. Now, why beta carotene as a source of vitamin A? You know that there are two types of vitamins. One is fat-soluble vitamins. Second group is your water-soluble vitamin. Now, when fat-soluble vitamin, these are A, D, E, and K. And water-soluble vitamins are B complex vitamins. Now, this A, D, E, and K, these vitamins are fat-soluble vitamins. So, if you take these vitamins in excess, they will be deposited in your body and cause the symptoms of hypervitaminosis. But if you use beta carotene as a source of provitamin A, it has been found that it is not harmful, and if it is in excess, it will not result in the symptom of hypervitaminosis. Then second pigment what I am talking about is the chlorophyll. You see that this chlorophyll, it has this is spiral, tetrapyrrole. In the center of this tetrapyrrole is magnesium ion, and this is phytol tail. If you compare with heme of hemoglobin, it is also made of this tetrapyrrole, but in the center, there are two differences. In the center, there is iron atom, and this phytol tail is not available. Now, there are several articles, there are several papers in the net you can search that this chlorophyll is also called blood of plants, and if you take sufficient of chlorophyll in your food, this chlorophyll is converted to heme in your body. How? Body, what it does, it actually cut this phytol tail, and magnesium ion is replaced by iron in your body. So now chlorophyll is converted to heme in your body, and therefore your hemoglobin level will be elevated. Now this is another picture where you can see that this tetrapyrrole is component of many other molecules: chlorophyll A, then heme, then cytochrome, then coenzyme, then vitamin B12. These are, so this tetrapyrrole is very excellent thing. Now this healing power of, so you can imagine that if you chlorophyll is entered in your body, its phytol tail is cut, and magnesium is replaced by iron, then it is converted to heme. Now, this chlorophyll, that means entering chlorophyll in your body will elevate your hemoglobin status and at the same time elevate the oxygen status of your body because it is a carrier molecule of oxygen. Now, this thing is utilized in the Chinese medicine. Actually, in Chinese, they have their own system of medicine like our Ayurveda. They are treating cancer by this chlorophyll treatment. How? They are actually using a term dampness for cancer and dryness for normal condition. So if a cell is cancerous, it is now established that cancerous cell cannot thrive in oxygenated environment. So if oxygen level in your body is enhanced, that means it can affect in some or better way with cancerous cells. They can fight with cancerous cells by providing good quantum of oxygen. So these in Chinese medicine, they are using this. I shall go for that, but before that, okay, I shall talk about that part again. Now, there is another component here, nutraceuticals. Now, you can see that it has two components, nutraceuticals. That means you are taking any food which is also acting as pharmaceutical. I think in inauguration, these algae were discussed, but one by one I am discussing the spirulina, then chlorella, then synodesmus, then dunaliella, then AFA. Spirulina chlorella is known to most of the students now because spirulina is blue-green algae. It is known as the superfood of the future because it has 60 to 70% protein, huge quantity of minerals, huge quantity of vitamins, phycoerythrin, cyanin, etc. Chlorella, it is a green algae. It has 50% protein. Synodesmus, it is a green algae. It has also a good quantum of protein. Then there are two others, Dunaliella and AFA. You see, here there is a photograph of one of the lakes. It is photograph one of the lakes, Sambar Lake, Rajasthan. You know this about this lake because from Sambar Lake, this salt is prepared and they are sold in the market as Sambar Namak from Sambar Lake. This is a hypersaline lake. Now, in this hypersaline lake, one algae grows. This algae is known as it is a chlamydomonas-like algae. You can see the photograph. It is actually Dunaliella. Now, this alga is a very curious alga, very wonderful alga, because it, in this condition of high salinity, actually in this saline lakes, what happens at the time of rainy season, salinity may drop or salinity may increase. This is going on. It is a continuous process. Now, to manage in this situation is actually very difficult because if a living cell is put in a saline solution, it will either get exosmosis or endosmosis. If it will get more water, it will burst, or it will get it will leave more water, then it will be squeezed. So it is a fatal for salinity fluctuations. This is a fatal for any organisms. But this Dunaliella, it is called an euryhaline organism. Now, what it means? Euryhaline means algae or organism that can tolerate wide range of salinity fluctuations. That means if the normality of salt is 5 and it drops suddenly to 5 or 2, then the alga or organism will be able to manage. Then how it manage? It actually converts its photosynthate into glycerol. At the same time, you can see that since this is a stress condition, it will store orange colored pigment beta carotene in its body. So in the next couple of slides, I shall be talking about this. What is the spirulina? I mentioned about the spirulina. I think spirulina is known to everybody. Spirulina capsules are available in the markets. Doctors are actually prescribing for the malnutrition or protein deficiency. In many other cosmetic products, they are available in the markets. And I have seen this the shampoos made of spirulina in the markets of Madras. In grocery shops, they are selling spirulina powder for the washing of hairs of the female. So this is very good. Spirulina, it is used in therapeutics for anti-skin cancer formulations, wound treatment, and promote skin beauty metabolism. Then used in beauty products, anti-wrinkling agent, anti-pimple cream, face mask, and high-protein shampoos. I mentioned about it. This is spirulina, and I hope Professor B.N. Verma, I have seen him in this lecture. He is one of the experts of spirulina, like Professor your Professor Puspa Srivastav, L.V. Venkatraman, Professor Gigi Bai. This. And then this is spirulina, how it is made. Now, this is spirulina tablet available in the market. Now, this is one glass of spirulina. Actually, one professor, she is known as spirulina lady. I actually respect him. She is actually student of Professor T.V. Desikachari. She is in Madras. She was in Calcutta sometimes back for a lecture with Professor Rumapal in Calcutta. There. So she told that Professor Pal, I used to take 1 glass of spirulina most often. So it may be that she is above 80, I hope, but she is still active. I may think that spirulina may be the secret of her activity or like that. She is associated with one of the very grand institutes in Madras, Merugappa Chittiar Research Institute, with Professor Seshadri. She is doing research on spirulina, I hope still. So this was about the spirulina. Now this is chlorella. You see cup-shaped chloroplast, green chlorella in Taiwan. In Japan, chlorella soaps are often sold in the market. This alga was famous to the students that it was sent to space for its utilization as a protein source or source of oxygen. But I don't know that whether it was success, but in another field, chlorella probably have been utilized. Then this 50% protein it has. Now, I should mention one fact. I think students have heard about one term vegan, V-E-G-A-N, vegan. Actually, these vegan are a particular class of people. They are actually not consuming any animal product, neither mutton, chicken like that, or neither in veg conditions, say for example, milk. They are not even taking milk. They are all taking food 100% vegetarian. Mostly they take fruits. So one of my friends, he is vegan. So he was with me. I asked him, you are taking mostly non-cooked or this fruits, etc. So do you face any problem? He told, yes, sir, I actually face a problem sometimes. But I asked, what problem? uh, he told that I face problem for some B complex vitamin deficiency. Then I told, what you do for that? You take a chemical substance? But no, I don't take chemical substances. I used to purchase chlorella or spirulina tablets from the Amazon. I used to take as food supplement, and in this way I am very okay. So this is the wonderful about these things. Chlorella tablets I mentioned. Then I mentioned about the Dunaliella, that it is an euryhaline organism and it is accumulating huge amount of beta carotene and huge amount of glycerol. Sometimes back I have seen one company, ABL Limited, probably it was Vizag or Andhra based company. They were of inspiration that they can produce commercial natural glycerol from this Dunaliella, but I hope it was not commercially successful, because I have not seen in news anymore. Then if we can use Dunaliella for beta carotene production or for glycerol production, it will be a natural source of beta carotene and natural source of glycerol. Then this I was talking about AFA, Aphanizomenon floss aquae. It is one of the blue-green algae floating. And most wonderful thing about this alga is that, this is the alga, that it contains 3 to 6% of chlorophyll, and it is said that it is the most concentrated form of chlorophyll in any organism, in any photosynthetic organism. This alga, it is also excellent source of protein, but in Chinese medicine, this AFA is being used in treatment of cancer. I mentioned that cancer, they are actually speaking that cancer is a form of dampness. So to overcome cancer, you have to supplement good amount of oxygen to your body by elevating substances which can carry good amount of oxygen. They will be helping to fight you with cancer. So they are treating this chlorophyll. Indirectly it is converted to hemoglobin. Then it is from that oxygen level of body is increased. This is their I told about this Chinese medicine, and you can wonder that there are certain other things that AFA actually increase the production and release of natural killer cells. And you know that this can improve your immune level. So this is also utilized in that system. Then I mentioned that algae, they have pigment. I mentioned about the beta carotene. I mentioned about the chlorophyll. I should mention about the blue-green algal pigments. One is phycocyanin, one is phycoerythrin. Now this phycocyanin molecule, now you can see it is one of the advertisements where you can see natural anti-cancer molecule phycocyanin. It is now available in India. So phycocyanin is now used in the treatment of cancer. What much successful, the time will say, but they are in use. Then phycocyanin, I mentioned about it. This phycocyanin is a wonderful molecule because it will help to facilitate the drawing together of amino acids, the building block of neurotransmitters. Now, you know that in our nervous system, the impulse is moving in electrical chemical in this way. Electrical impulse is moving, and when it is moving through a cytosol or cell junction, there certain chemicals, neurotransmitters are actually used for chemical transmission. Then these neurotransmitters are actually made from certain amino acids. These are actually phenylalanine. The spirulina or phycocyanin, they elevate this phenylalanine level, and indirectly they help to improve the condition of neurotransmitters in your body. Now, I was talking about these all aspects, that how this phycocyanin is actually being used for the improvement of your oxygen level, or improvement of your immune level, or improvement of your phenylalanine level. And if the phenylalanine level, or if this neurotransmitting activity is improved, then it could be used in the treatment of Parkinson's disease, Alzheimer's disease, and multiple sclerosis, and many other diseases. So this is one of the things which is used. I have given a reference. This is Professor Catcher James. His article, several articles, he is a doctor, several articles are available in the net. So if students are interested, anybody can interested, they can surf the net and they may get it. Now, I was talking about beta carotene. So I can come back from that beta carotene actually is one of the molecules which is acting as provitamin A. Second activity of the beta carotene is that it is a very good antioxidant. Now, before taking into account what is antioxidant, you should first think about the oxidant activity. What is oxidant? Actually, there are several things. Suppose if you use one water molecule is almost a neutral molecule. If you break it, you will find it one hydroxyl ion and one hydrogen ion. Hydrogen ion has a plus charge, hydroxyl ion has a minus charge. Now, these are actually free radicals. The hydrogen ion will try to get in one electron, and hydroxyl ion will want to get one proton. So they are in excited state, and they will move here and there to get electron or get proton. So they are actually disturbed molecules. If any molecule or any free radical, it is disturbed, it will go, and what it will do, it will go and disrupt, it will it will actually disturb the lipid bilayer of cell membrane, and if your cell membrane is damaged or being destroyed or being disturbed some way, that means your skin will go in a worst condition. Ultimately, if your skin is not better, then only additional cosmetics will help to feel you better. Otherwise, pimples or many other skin rashes will be the result of that. So, to scavenge these free radicals or oxidants, now the antioxidant concepts are available. What antioxidants actually do in next slide, you will see that antioxidant, it react with free radicals and make the molecule stable. That means they have the scavenging activity. Sweeper जैसे करते हैं कि गंदा साफ कर देते हैं, ये क्या करते हैं, एंटीऑक्सीडेंट्स जाके फ्री रेडिकल्स को इनएक्टिव कर देते हैं, एंड इन अनदर वे ये आपके स्किन तक इनको पहुंचने ही नहीं देते हैं, या बॉडी में किसी भी तरह की केमिकल डिस्टर्बेंस करने नहीं देते हैं. मैं बहुत दिन पहले एक टीवी शो देख रहा था, तो उसमें मैंने देखा कि एक बहुत फेमस एक हॉलीवुड एक्ट्रेस थी. वो हॉलीवुड एक्ट्रेस, उसका नाम है, बहुतों ने सुना होगा, जेनिफर लोपेज. तो जेनिफर लोपेज को इंटरव्यू कर रहा था, एक लेडी इंटरव्यू कर रही थी. तो जेनिफर लोपेज को इसने पूछा कि अच्छा मैडम, आप अपना जो ब्यूटी है इसका आपका सीक्रेट क्या है, आप कैसे मेंटेन करती हैं. तो उन्होंने 3-4 बातें बताई थी. पहली बात उन्होंने ये बताई कि मैं तो रोज योगा करती हूँ, दूसरी बात उन्होंने बताई कि मैं कम से कम 3 लीटर पानी रोज पीती हूं, क्योंकि पानी जो है वो आपके शरीर की गंदगी को धो के बाहर निकाल देता है। और तीसरी बात उन्होंने बहुत इंपॉर्टेंट बताई कि मैं उन सारे फलों को खाती हूं जो काट देने से, फल को काटने के बाद उसमें किसी तरह से रस्टिंग हो जाती है। देखिए रस्टिंग जो है ये तो बहुत क्रूड टर्म है। आप एक बनाना काट के रख दीजिए, एक आप एप्पल काट के रख दीजिए, या एक आप इसको क्या कहते हैं, हम लोग यहाँ प्यारा बोलते हैं, आपके यहाँ अमरूद बोलते हैं, अमरूद काट के रख दीजिए, एक पपीता काट के रख दीजिए। देखिए कि उसके ऊपर एक रस्टिंग की तरह का वो पड़ जाएगा कुछ देर के बाद। तो ये एक्चुअली इनडायरेक्ट एक सिंबल है कि ये ऑक्सीजन ट्रैप करता है और उस तरह से कुछ रस्टिंग हो जाती है, इस तरह की बातें होंगी। लेकिन इसका एक इनडायरेक्ट इसका एक सिम्टम है कि इन कंपाउंड्स, इन सब्सटेंसेस में इन सब्सटेंसेस में एंटीऑक्सीडेंट्स का लेवल अधिक होता है। तो वो जो है बाद में मैंने एक्सप्लेनेशन इसका पढ़ा कि एक्चुअली इन सब चीजों का ये इनडायरेक्ट एक टेस्ट है कि इनमें एंटीऑक्सीडेंट्स ज्यादा है। इसका मतलब है कि इस तरह के फल आप अगर खाएं, इस तरह की चीजें आप खाएं, तो आपके बॉडी में एंटीऑक्सीडेंट्स के लेवल अच्छे होंगे और इससे आपको स्ट्रेस को कॉम्बैट करने में मदद मिलेगी। तो ये चीजें हैं बहुत इंटरेस्टिंग जो एंटीऑक्सीडेंट्स के मैंने बताया। तो आपका कैरोटीन, बीटा कैरोटीन या बहुत सी चीजें हैं वो एंटीऑक्सीडेंट की तरह काम करता है। तो आपके स्किन को भीतर से भी, आपके शरीर को भीतर से भी दुरुस्त करने में ये मदद करता है। डुनलिएला के विषय में मैंने बताया। एक और चीज है यहाँ, ये देखिए, ये है एस्टाजेंथिन। एक और पिगमेंट है जो ग्रीन एल्गी में ज्यादातर मिलता है, वो है एस्टाजेंथिन। और एस्टाजेंथिन एक पर्टिकुलर एक एल्गी है जिसमें बहुत ज्यादा होता है। एक है हिमाटोकॉकस, वो स्नो हैबिटेट में नॉर्मली ग्रो करता है। उसके विषय में मैं बाद में आपको थोड़ा सा दिखा रहा हूं। ये है हिमाटोकॉकस। ये हिमाटोकॉकस ये स्नो एल्गी की तरह इसको बोल सकते हैं, और इसमें ये एस्टाजेंथिन काफी एक्यूमुलेट ये करता है। तो इंटरेस्टिंग बात यह है कि ये एस्टाजेंथिन, मैं मद्रास में प्रोफेसर, ये आपके देखिए कि बाद में फोटो दिखा रहा हूं, ये है प्रोफेसर आर रेंगास्वामी। इनको मैं बोलता हूं एस्टाजेंथिन प्रोफेसर, क्योंकि इन्होंने एस्टाजेंथिन पर और इन सब पर काफी रिसर्च किया है। और अभी शायद मलेशिया में कोई मल्टीनेशनल कंपनी है, उसके ये शायद मैनेजिंग डायरेक्टर हैं, और वहां ये आपके हिमाटोकॉकस की जो कल्चर है उसको देखते हैं। अच्छा, ये अभी इनकी ली हुई फोटोग्राफ है 1-2 साल पहले की, और आपको देख के इनका एज क्या लगता होगा? 40 साल के आसपास होंगे, 40-42 साल के आसपास होंगे। तो प्रोफेसर चौगले भी हैं, बाकी लोग भी हैं, इन्होंने इनको देखा होगा। ये इनकी एज एक्चुअली 65 प्लस है, 65 के ऊपर है ये। अभी मद्रास यूनिवर्सिटी से वहां से वो रिटायर करने के बाद ये अभी वो मलेशिया में ये करते हैं। तो उन्होंने हमें एक बात मजे में बताई थी कि प्रोफेसर, इफ यू टेक एस्टाजेंथिन, few milligrams of एस्टाजेंथिन, you will get never old. I don't know ये जो बोल रहे हैं ये कितना सच है, क्या है, लेकिन उनके चेहरे से देखिए, उनके बाल अभी भी काले हैं। आप उनसे मिले तो 67 के एज के होंगे, लेकिन अभी ऐसे ही हैं, और ये ग्रैंडफादर भी बन चुके हैं। और एक इंटरेस्टिंग बात आपको सबके लिए बड़े बड़े मजे की बात एक बताऊं। इनके जो टीचर हैं प्रोफेसर एन आनंद, यहाँ आए थे, हमको एक बात कह रहे थे कि आप जानते हैं कि ये इनके घर में केवल एक ही किताब है, वो है श्रीमद् भागवत गीता, और घर में कोई किताब नहीं रखते हैं। जो इनका जो काम है वो लेबोरेटरी में ही होता है। तो खैर ये बात है। इसमें मैं आपको एक और प्रोफेसर के विषय में बताऊं, प्रोफेसर ताजुद्दीन। ये भारतीदासन यूनिवर्सिटी, वहां पर इनका अपना एक बहुत बड़ा प्रोजेक्ट चल रहा है माइक्रो एल्गी के ऊपर। तो इन्होंने एक, देखिए, ये अपने वाइस चांसलर को 12 जुलाई 2016 को उन्होंने मेरे को ये फोटोग्राफ भेजी थी कि अपने प्रोफेसर को ये जो है, अपने वाइस चांसलर को उन्होंने एक बर्थडे केक बनाया है। और देखिए, इस बर्थडे केक में इन्होंने जो कलरिंग की है, देखिए ये ब्लू है, ये फाइकोसाइनिन से कलरिंग की गई है, और ये जो है पिंक कलरेशन है, ये फाइकोएरिथ्रिन से कलरिंग की गई है, और मिक्स्ड कलरेशन है, ये फाइकोसाइनिन फाइकोएरिथ्रिन दोनों मिला के कलरिंग की गई है। तो अब कोई तो प्रोफेसर इतनी उसको बुद्धि होगी कि ऐसी कोई टॉक्सिक चीज अपने प्रोफेसर को जरूर नहीं खिलाएगा, अपने वाइस चांसलर को। तो देखिए जो ये एक पॉजिटिव चीज है कि पिगमेंट्स जो है दे आर बीइंग यूटिलाइज्ड फॉर दिस, एंड फॉर द astaxanthin or these pigments, somewhere I have seen that that at the time of aging, repeating the words of Professor Rangaswamy, once I have seen one article, there it was said that these pigments, what they do, actually what happens at the time of aging, this your DNA, the end of DNA chain, end of DNA, it actually exists in hairpin loop. Then its hairpin loop is open at the time of aging. Now these pigments actually they actually keep the these things hairpin loop intact. I think I'm actually at the end of the lecture. So these were the things what I was to say. Now this is chlorococcum, another alga. Now future food. I was telling about some food items. This is Dr. Soul Superfood. These food are actually available in the market. They are actually nutraceuticals. Most of this food is made of some algae. They are certain macroalgal food. You see, this is alba, it is called suletus. Then porphyra, it is called nori, and this nori is has a several billion-dollar industry in Japan, Taiwan, Korea, and certain parts of United States and Canada. Now, this is a Gulf of Mannar cultivation of Eucheuma. This is one of the seaweeds which we are cultivating in our country. Then this is one of the seaweed dishes. This is one. Then you can see these are the algal products today in the market. You can see, देखिए यहाँ जो जेम्स ये भी बने हुए हैं, several type के ड्रिंक्स भी बने हुए हैं, फेस मास्क भी है, और तरह तरह के देखिए केलॉग्स का जो क्रिस्पी आपका ये आता है, कॉर्नफ्लेक्स आता है, उसमें भी दिया हुआ है। तरह तरह के प्रोडक्ट्स हमारे देश में भी अवेलेबल नहीं है। लेकिन जो बात यहाँ इस लेक्चर में मैं बताना चाह रहा था कि तरह तरह की कॉस्मेटिक्स जो अवेलेबल हैं वो नेचर इको फ्रेंडली कॉस्मेटिक्स, बायो कॉस्मेटिक्स एल्गी से अभी बन रही हैं, और अभी लोग इसको यूज कर रहे हैं, जो कि आपको स्किन को बेटर रख सकती हैं। केवल बाहर से नहीं बल्कि आपके ग्लैमर को मेंटेन करने के लिए ये क्लोरोफिल है या बीटा कैरोटीन है या फाइकोसाइनिन है, फाइकोएरिथ्रिन है, आपको ये भीतर से भी आपको ये हेल्दी कर सकती है। तो ये था लगभग जो मैं कहना चाहता था लेक्चर में। अब थैंक यू फॉर लिसनिंग my lecture to all of you. So this is the end of my lecture. I shall be happy to get the answer. If you wish any answer, I shall be happy to reply from the audience, if any.
Thank you, Professor Khatri, for a nice lecture. Now the lecture is open for discussion. Anyone who wants to ask question, please ask.
Dr. Keshri, thank you very much. I am really
अच्छा, नमस्ते सर। मैंने आपको देखा, मैंने वहां आपको देखा, आपने आपसे फिर विश नहीं हुआ आपके। हाँ सर, बोलिए सर।
I heard your lecture, very nice. You have updated the knowledge of the use of the algae, but I think we have collected enough data on the utility of algae. Now, what is important that this use of the algae for a common people, it should be brought to the knowledge of the common people how to grow it, how to use it. I remember Gigi Bai when spirulina was discovered, and it was found that protein is there. So Gigi Bai started taking this spirulina in the bread of his common use. So what's required? You are from Bengal. Yeah, I'm from North Bihar. Areas are very rich in the water pockets, and these water pockets are get beautified and giving the luxurious growth of autotrophous algae. So what is required? If these areas are developed as a farming of the algae, and these useful algae are brought to these places, and the people are get trained to cultivate it, to use it in common things. That will be more beneficial. We have lot of algae have been discovered with the good product. So what is needed in India, that it should be the people should be trained to grow it in the village areas, to use it, to practice it in the common food. So thank you very much. That part should be.
सर थैंक यू। मैं एक दो बात यहाँ थोड़ा सा हाँ सर हेलो हाँ सर यहाँ आपसे एक दो बात मैं शेयर करूंगा सर। हाँ सर थैंक यू सर। यहाँ एक दो बात शेयर करूंगा। हुआ क्या सर, यहाँ कुछ साल पहले मुझे याद है कि 5-6 साल पहले प्रोफेसर करगुप्ता भी जानते हैं कि यहाँ मेदिनीपुर में इन्होंने स्पिरुलिना कल्टीवेशन स्टार्ट किया था। तो हुआ क्या कि उनको ठीक से मार्केट नहीं मिला है। एक तो ये बात है। दूसरी बात है सर, एल्गी को पॉपुलर करने के लिए थोड़ा सा इसको बिस्किट में या बहुत सी चीजों में मिला के, जैसे अब जीजी भाई की बात कह रहे थे, इसमें खाना पड़ेगा क्योंकि सर हाँ सर क्योंकि बंगाल में है क्या, लोग स्वाद के पीछे ज्यादा भागते हैं सर सारे जगह ही भागते हैं, तो उनके जीभ के पास हमको किसी न किसी तरह से ले जाना पड़ेगा सर। हाँ हेलो
at least for we people belonging to the North Bihar, these are the handful is going useful and practicing its use in the daily food. So that is due, and that should be started from psychologists like you, me, Chaudhary, and all that. So I'm too old now. I'm touching 82 years. So
नहीं सर, अभी तो कुछ दिन पहले आपने लेक्चर भी दिया, उसमें बायोडाइवर्सिटी वाले में बड़ा अच्छा लेक्चर दिया सर आपने आपने उधर मद्रास में भी अभी लेक्चर दिया। अभी तो बहुत एक्टिव है सर आप
After a long time I could see you, and
thank you sir.
Very happy to have given a very nice lecture.
Thank you sir. Thank you sir. Thank you sir.
Thank you very much.
Thank you. Yes.
Anyone who wants to ask any question? प्रोफेसर केसरी से क्वेश्चन पूछना चाहते हैं? Thank you, Professor Kesri, for a nice.
Thank you, sir. Thank you for this opportunity. Thank you very much.
लेक्चर दिया.
Thank you, sir.
एक न्यू कॉन्सेप्ट algae in glamour के बारे में बताया.
हाँ sir.
The entire University Department of Botany is very grateful to you, sir.
Thank you, sir. Thank you. Thank you, sir.
Okay, sir.
Thank you. Thank you. Thank you, sir.
Okay. Nice. Break the neck. Break the neck. Break for 15 minutes. Hello, audience. there is a break for 15 minutes, and after 15 minutes we are joining again. That is tea break.
Professor Shahu, it is already about one. And what about the session which I have to share? Pramod, are you hearing?
बोल रहा है दादाजी क्या हुआ
नहीं चल रहा है
Hello. This is the time for second session. In the second session, local participants will deliver their lecture, and amongst the research scholar, Dr. Swati, PG Department of Botany, LNME. So I invite Dr. Swati, PG Department of Botany, to deliver her lecture. Topic of her paper is Record of New Taxa of genus Oedogonium. So, Dr. Swati, please come.
Yeah, hello. Hello, am I audible?
Yes, audible.
Hello, Swati.
Yeah, a very warm. Yeah, may I start, sir?
No instruction. Oh, okay.
A very warm good morning to all of you, all the delegates who are joining here on the National E-Seminar on Algae and the Human Welfare. All the delegates from the Department of Botany and the Biotechnology, and from the all over the India who is joining here on the offline or online mode. This is Swati Kumari. Hello. Am I audible, sir?
Yes, audible.
Yeah. My slide is visible or not, sir?
Yes, visual.
Yes. Yeah. Today I'm going to present the record of new taxa of the genus Oedogonium from the North Bihar district, which I have done the work. Actually, I have worked on mainly on the order Oedogonians of the North Bihar during my research project. So this is the part of that, and I'm going to present on all the delegates that the the new taxa has been identified during my research, which comes under the order Oedogonians. Here I am going to show the systematic position of Oedogonians, which comes under the division Chlorophyta, class Chlorophyceae, order Oedogonians, and the family Oedogoniaceae. The Oedogoniaceae is the family which compiled only the three genera, that is the Bulbocate, Oedogonium, and the Oedocladium. The order Oedogonians was established by G.S. West. It comprises only a single family Oedogoniaceae, and the family Oedogoniaceae is constituted by D. Barry in 1854. The genus Oedogonium was created by Link, which is particularly the aquatic taxa of the green algae, which is the thalloid, which is mainly the unbranched. There is not a single taxa has been identified which is branched, and the holdfast, that is the holdfast is the part of the thallus which is beneficial for the attachment of the thallus to the substratum, and the seta is absent here. It is mainly present in the other genus like a Bulbocate in the same family. Here are some key characteristics of the Oedogonium on the basis of which anyone can identify this genus. That is the presence of holdfast, the capsule, the vegetative cells shape and size, the shape of the Oegonium, ornamentation of oospore wall, the presence of the dwarf male, and the condition Ganendrosporus and Idioendosporus. So here I want to explain the condition Ganendrosporus and the Idioendosporus. Actually, endospore is a cell which develops the dwarf male in the thallus. And if this endospore is present on the female plant, then it is known as the Ganendrosporus. And if it is present on the male plant or other plant other than the male plant, then it is known as the Idioendosporus. And the presence of Antheridium, that is the male part, and the Androsporangium, that is the identifying characteristic of this genus. Here in this paper, I am going to describe the 3 new taxa identified. That is the Udogonium tiffany ac variety Indiana variety norm, the Udogonium gonzalvesi Islamian sarma variety Desica charai variety norm, and Udogonium caruguptai species norm. So here I am going to present one by all of the 3. The Udogonium tiffany oak variety Indiana variety norm, which is mainly the Macrandus species. It means the Macrandus stands for the if the taxa which has the male and the female part are developed on the different thallus, then it is known as the Macrandus species. So it is a heterothallic. Here I found the vegetative cell cylindrical and the suffultory cell cylindrical. Ugonium were found in the 5 to 6 row, which was ovoid in the shape and broadly pyriform ovoid. This specimen was poriferous. It means the pore is present in the ogonium, and the position of the pore was the superior. Oospore was the ooglobose or to the ovoid, and which was not filling the ogonium longwise. Spore wall 3-layered and the medial wall scorbulate. I will show in the next slide the ornamentation and all the things that I am describing here. The antheridium was found in the 4 seriated, and the spermatozoid were the 2, whose division was horizontal. Here I am showing the next in the slide. Here you can see the antheridium in the 4 rows, and here you can see the division is the horizontal in the antheridium, and here you can see the ogonium in the 4 to 5 rows present here. This is a camera lucida drawing which I have drawn during my research. This is the ascorbulation in the oospore wall. You can easily see seen here, and the antheridium shape and the size. Here I can show you the ascorbulation in the ogonium wall. Is this visible? Hello? Hello? Is this visible?
Yes.
Yes. Here I have found this specimen. I have collected the specimen mainly from the Kalyanachar from the district Darbhanga in the December, and this specimen was found growing on an angiospermic plant as an epiphyte, where the pH was recorded 6 and the temperature were the 19 degrees Celsius of the habitat. Why I considered it as a new variety? Because when I compared this specimen to the already identified specimen, I found is a close relation with the Odogonium tiffany. But here is some differences that demarcated it from the other varieties, that is a ovoid, broadly pyriform ovoid shaped ogonium, and cylindrical suffultory cell, and less broader vegetative cell and antheridium were the separate from the present specimen. So it was very close to the forma tiffany, but on the basis of the above differentiation, I considered it as a new variety and named it as a Odogonium tiffany variety Indiana variety norm. The next taxa is the Odogonium gonzalvesi Islami sarma variety Desica charai variety norm. It is an nanandrous idioendosporous species, variety. And nanandrous means there is a presence of a dwarf male on the suffultory cell. Here I have taken the micro photography through the slide, but there was a presence of a dwarf male there, but when I took the photographs, it displaced from there, so I have mentioned it here. Here you can see the 3-layered wall of the ogonium, and here you can see the ornamentation of the oospore wall, which I have mentioned here. Ogonium suffultory cell are inflated. Ogonium in the 2 or subovoid to the quadrangular globose. This specimen is also a poriferous, and the pore was found to be medium. It means it is getting ruptured from the middle of the ogonium. Spore wall is clearly visible, the 3-layered, and the longitudinally costed anastomoizing at few points. Type of ornamentation has been found. Dwarf male were the present on the suffultory cell. Here you can see the dwarf male is what's present like this, but it get displaced after when I have taken the micro photography. So this is a actual ornamentation that I have seen under the microscope. I have drawn it on the laboratory. This specimen has also collected from the Darbhanga district, Kalyanachar, in the month of December, and was growing as an epiphyte there in the pH of 5 and the temperature 96. It is considered as a new variety because in the original description, it is the idioendosporous condition, which has been shown doubtful. Means it was not clear it is a gynandro or idioendosporous, so it was not clear there. Moreover, the position of pore was not found in this specimen. So in the present study, the pore is the median in the position, but in the actual description, there was the position of pore was not clear. So because of the capitulate cell and the median pore of Ugonium, it differs from the other variety of a striatum and come closer to the Gonzalvesi islamicerma. So on the basis of this, I have compared this specimen to the other varieties or the forma. So I came closer to the Odogonium Gonzalvesi islamicerma, but the presence of making in between the costate make it differ from the Gonzalvesi too. So it is considered as because the ornamentation is the genetical characteristic of an organism. So it is considered as a new varieties of this species, and its name given in the honor of Professor T.V. Desikachari, as noted Indian phycologist. So I named it as a Odogonium Gonzalvesi islamicerma variety Desikachari variety Nov. The next one is the new species that I have identified during my research. It's also an anandrous species, Odogonium cargoptai species norm. Here I found the vegetative cell cylindrical and the inflated suffultory cell. Ogonium were found single and subglobous to ellipsoid. And the spore, it is also a poriferous specimen. So it is a pore was found to be superior. The oospore globose, and which was not filling the Ogonium completely. The spore wall ornamentation, you can see here the scorpulation on the side wall of the oospore. Here it is clearly visible, and you can say I have found this and dwarf male was found almost straight and situated on the sulfuratory cell. This specimen was also collected from the Kuseswarasthan, Darbhanga district from the month of March, and it was also growing as an epiphyte. The median wall of spore appears scorbiculate, continuing with the spiny longitudinal costa. The space between the costae seems annulated. Such unique ornamentation has not been detected from any other scientist in this anandrous group of algae. So it is named as an Odogonium cargoptai species norm in honor to my teacher and the mentor, Professor A.N. Kargupta. So this is the, I'm compiling my presentation here. So thank you so much for giving the opportunity to present my work here in the section. Thank you so much. Hello.
Thank you, Swati.
Thank you so much, sir. Yes, thank you. Pranam, sir. Yes, sir.
I'm B.N. Verma speaking.
Yeah, good morning, sir.
I heard your lecture. You have made
yes,
a very good contribution. You belong to
Thank you so much, sir.
Kar Gupta, and it's a very devoted work. But I tell you very well, frankly,
yes, sir.
The morphology is subject to ecological variations.
Yes,
although the Udogonium, you must be knowing, it has been advised to give a special status of the class Udogoniophile or all, but it is only because of its very interesting vegetative division and reproduction. So these are so distinct that it needs isolation as a distinct. But I tell you, when you will go in the cytology, deep into the cytology of the Udogonia.
Yeah. Actually, I had a plan, sir. I had a plan to go for cytology.
That's why I have done a few cytology of a few species. Although Udogonians have gone to a very high specialization in regard to the vegetative reproduction and sexual reproduction. But when you will go in the cytology of the Udogonia, you will find the main feature of the algae, what makes it distinct from the higher plants, that the plants are haploid. Secondly, the plant polyploidy is not a normal phenomenon of the algae.
Okay.
So that phenomenon has been retained by the Udogonians. There is, unlike the charophytes, unlike the cladophorals, you will not get any polyploid series.
Okay.
But chromosomes are at par with the charophytes, at par with the cladophorals. The chromosomes are well defined and well of good size, well distinct centromeres and all that. But when you will go deep in the level of the genus, unlike the other group of the algae, where the genera are distinct with its distinct chromosome set, that you will not find in the Udogonium. That lacks. There is no cytological differentiation, apparently. That's why the people are going on the estimation. Thank you very much.
But thank you so much, sir, for your suggestion. I will take it for my near future.
I will go into the cytology of and will find very interesting things.
I will try. I will take your suggestion for future. Thank you so much, sir.
Thank you. Thank you. Thank you.
Paper. Now, the second speaker is Dr. Ankit Kumar, MRM College, Darbhanga, Marwari College, Darbhanga, and the topic of his research paper is use of a halo-tolerant chlorophyte as anti-cancerous drug resource, a nanotechnological approach. So, Dr. Ankit Kumar, please present your paper.
बोलिए। बोलिए स्लाइड दिखेगा तब ना बोलेंगे।
A very good afternoon to all. Am I audible? Am I audible? Use of halo tolerant. A very good afternoon to all. Today's topic of my presentation is use of a halo tolerant chlorophyte as anti-cancer drug resource, a nanotechnological approach. As we know that cancer is one of the major cause of death worldwide. According to World Cancer Report 2014, 60% cancer patient from Africa, Central and South America, and 40% from other countries. Most of the cancer treatment include classical chemotherapy, radiotherapy, and surgery method. These standard methods are toxic and have several side effects on normal cell with limitation of their use. So there is an urgent need to develop such a method which are non-toxic, eco-friendly, and acceptable by the normal people. Please next. According to Craig et al. 1997, 60% anti-cancer drug from natural compound and 40% from synthetic origin. Next. These are some anti-cancer drug from microalgal and cyanobacterial origin. Cryptophycin 1 from nostoc species. It is used for cancer treatment and is launched in market by Merck Private Limited. Nigricanocytes A from TDB and brintuximab vedotin 63. Green carbon nanotax and chlorella gold nanorod hydrogel for treatment of breast cancer. Please next. Why nanoparticles? Nanoparticles synthesized by biomolecule in comparison to administration of free bioactive molecule possess several advantages like accumulation of drug at target site, better drug delivery by increasing solubility of bioactive molecules, increased half-life of drug in circulation. दिख नहीं रहा मुझे इसमें, मैं यहीं से बोलूंगा. Accumulation of drug at target site, better drug delivery by increasing solubility of bioactive molecules, increased half-life of drug in circulation, stable and constant drug release, decreased drug resistance mediated by efflux pump, protection of bioactive component and its bioactivity in serum-rich environment. So microalgal based nano formulation with efficient anti-cancer activity may be alternative to expensive and toxic cancer therapy. Dunaliella salina is a unicellular wall-less chlorophyte isolated from hypersaline Sambar Lake, Rajasthan, and not known to have any toxicity to human cells. Next. In present study, I have optimized the process of synthesis of silver nanoparticle using aqueous extract of Dunaliella salina. I have optimized the process of synthesis like sunlight exposure time, AED inoculum dose, and AgNO3 concentration. Next. At optimum conditions, synthesized nanoparticle were characterized using transmission electron microscopy. These are the 10 images at different magnification which show synthesized nanoparticle were nearly spherical in shape. Figure E shows the SAED pattern, selected area electron diffraction pattern, which show the crystalline nature of nanoparticle. Figure F showed the size distribution histogram, which show the particle size range between 1 to 30 nanometer with average size of 16 nanometer. Next. Further crystalline nature were confirmed using XRD analysis. Figure B show the FTIR analysis of aqueous extract of Dunaliella salina and synthesized nanoparticle. This reduction in peak intensity indicate that involvement of various functional groups present in aqueous extract of Dunaliella salina were used in the synthesis of nanoparticle. This is the atomic force microscopy analysis. we saw the surface roughness profile of the synthesized nanoparticle. Next. At optimum conditions, synthesized nanoparticles were screened for their anti-cancer activity on breast cancer cell line MCF7 and normal breast epithelial cell line MCF10A. This figure shows to see the live and dead cell ratio. We perform the calcine PI dual fluorescent assay. Calcine AM is a hydrophobic dye which enters in the live cell and break into fluorescent calcine by esterase enzyme. Am I audible?
Yes, yes. But your slide is not visible. Your slide is not visible, sir.
टेक्निकल दिक्कत आ रहा है सर. Am I audible?
Yes, you are audible now.
No, okay. You just go on. Your slide is audible, visible, and your voice is also audible. So go on.
बहुत दिक्कत हुआ टेक्निक बीच में गायब हो गया था ना, तभी तो बंद हो गया बोलना. बोलिए बोलिए बोलिए
बोलिए. Hello, am I audible? To see the anti-cancer activity of synthesized silver nanoparticle, we have performed dual calcine PI dual fluorescent assay. Calcine AM is a hydrophobic dye which enters in the live cell and break into fluorescent calcine by mitochondrial esterase enzyme. PI enters in the cell with damaged membrane and bind with nucleus and give red fluorescence. Here both cisplatin and silver nanoparticles with the cells with increased number of cells with PI. We saw both cisplatin, which is a known anti-cancer drug, and synthesized silver nanoparticle kills the cancer cell line MCF7 as here we can clearly see the increased number of cells with PI fluorescent. Next. Here both cisplatin and silver nanoparticle does not affect the normal breast epithelial cell line MCF10A, but cisplatin at higher dose also kills the normal cell. Here at 50 mg per ml at 48 hours, we can clearly see the cells with PI. Next. Cancer, as we know that cancer cells survive by inhibition of progression of apoptosis. To see the level of apoptosis, we perform the Annexin PI assay. Here both cisplatin and silver nanoparticle kills the cancer cell via induction of apoptosis. Here at 48 hours, more than 70% cells are either in early apoptosis or in late apoptosis. Next. Here in case of normal cell line MCF10A, both cisplatin and silver nanoparticle does not affect the level of apoptosis. Next.
आपको
I don't know whether Ankit Kumar has completed. Ankit had completed his lecture.
Hello?
Neither sound nor your picture is coming up. What is
Sorry for technical disturbance. Conclusions. The Nalliela salina can act as a source of green synthesis of silver nanoparticle. ED synthesized silver nanoparticles show selective killing of cancer cell line by induction of apoptosis and are not detrimental to normal cell lines. A comparison of known anti-cancer drugs, cisplatin and green synthesized silver nanoparticles against NCF10A cells, normal cells, and cancer cells, NCF7, revealed silver nanoparticle may be used as potent anti-cancer agent. Further identification of active molecule of Nalliela salina on the surface of silver nanoparticles would help in elucidation of novel anti-cancer agents and in designing of silver nanoparticles with vast improved anti-cancer efficacy. Thank you.
For your nice presentation. Anyway, some technical difficulty arises.
Can I speak to Ankit? Can I speak to Ankit?
Yes, you can speak to Ankit.
I want to know, Ankit, I want some question from Ankit Kumar. Ankit Singh. Is he hearing? Is he hearing?
बोलिए आप यहाँ से सर। हेलो, आवाज आएगा तो आवाज आएगा, बोलिए आराम से बोलिए।
Yes, sir.
Can I speak to Ankit?
Yes, sir. Yes, sir.
Ankit, you have done a very good piece of work. You have done a very good piece of work, especially I think it's good to note where you have done it in Mithila University laboratory or elsewhere.
No, sir, in BHU. In BHU, sir.
Oh, so
in BHU and anti-cancer aspects were done in Indian Institute of Toxicology Research Center, Lucknow.
Oh, that's why I was wondering how this type of work was performed in LNMU. I was just very nice. But what I suggest, Mr. Ankit, you have all what we are doing, exploring the potency of the algae under the laboratory condition. Now time is mature that you told these algae, BDA are having this potency and you can manipulate by altering the situation of growth and all that. So I mean to say, when you will visit the North Bihar, you will get a lot of water pockets full of growth of the big green algae. If that situation is exploited for growing these economy, the algae of eutrophic BG of blue-green algae member. In that situation, lot of benefits we can just fetch what you have gathered knowledge on the potency of the algae. So I misunderstood. I thought that you worked it in the Darbhanga Mithla University. Thank you. You have done a very good.
Thank you, sir. Thank you, sir.
What I want to say now, these types of work should be bring from laboratory to the land, to the field, so that its maximum benefit can be felt and utilized in the interest of common people. And thank you, very nice.
Thank you, sir. Thank you, sir.
How did you, one thing more, how did you manipulated the growth condition to optimize the cancerous substance anti chemical in that algae you work? How you manipulated the growth conditions?
Sir,
definitely you must have changed the growth conditions, the ecological situation to optimize. How did you optimize the cancerous elements of the algae? How did you optimize?
Kumar is a newly recruited teacher from VPSC and posted in Marwari College, Darbhanga. He has done his work at BHU.
So was it done in phytotron? Was it done? It is only the phytotron because the ecological situations can be controlled under phytotron. So where he manipulated the ecological to induce that to optimize the cancerous elements of the algae. How did he optimize?
Sir, hello. Am I audible?
Yes.
Sir, Danalina salina can survive in harsh environmental condition and produce secondary metabolite. As we know that most of the anti-cancer drug from natural resources were produced. That is a secondary metabolite. So I have synthesized the Hello.
How did you optimize? How you manipulated the content? You just said that you optimized the certain.
Sir,
how optimization
optimization of growth condition. Hello.
Yeah. I mean,
sir, hello. My portion is sir synthesis of nanoparticle.
Hello. Now, a student of third semester, Department of Botany, LNMU, will speak on role of algae in the 6th mass extinction. So, please, Keshav Kumar Misra, come and present your paper.
Good afternoon. Myself Keshav Kumar Mishra, a student of PG Department of Botany, Lalit Narayan Mithila University, Darbhanga. My topic is role of algae in the sixth mass extinction as well as its role in human welfare. Earlier, top 5 extinction has happened in different periods. The first was Ordovician Silurian extinction that was 440 million years ago. And second one was Devonian extinction that was 365 million years ago. And the third was Permian Triassic extinction 250 million years ago. And then Triassic Jurassic extinction 210 million years ago. And the fifth was the Cretaceous Tertiary extinction that was 65 million years ago. After the 5 mass on the Earth that were caused by meteorite impact, volcanism, and large-scale climate change, several scientists predict that beginning of the 6th mass extinction. In this scenario, it is humankind that is causing the mass extinction with the illegal trade in wildlife as one of the most important hazards to wildlife species. Many endangered species are being affected by the wildlife trade, both legal and illegal, which poses a threat to human health, is a major cause of species extinction and is eroding the ecosystem services that are vital for our survival. Now, the role of algae in the sixth mass extinction in this coronavirus pandemic, all over the entire world are suffering from a huge disastrous natural phenomenon that is caused by the COVID-19, SARS-CoV-19. But algae also play a major role in this scenario when we are discussing about the sixth mass extinction. Today, just about anywhere there is water, there can be toxic algae. The microscopic plant usually exists in a small concentration. But a sudden warming in the water or an injection of dust or sediment from land can trigger a bloom that kills thousands of these poisonous shellfishes or even humans. Suppose we are going to discuss about the Mithilanchal region, there are a lot of ponds. The Mithilanchal is known as the area where ponds, there is a large amount of ponds. But the conditions are very bad because there is a lot of nalas are go through there, and so due to the hazardous chemical compound present in that pond, we can say that it's also play important and critical role to extinct of the so many species that present in the ponds, especially in aspect to fish. So if we through theories of mass extinction to different types of general, some unanswered question, example, and impact. How does that the cause species to go extinct? Look, when we are discussing about the coronavirus, the people says and scientists says it comes from China because the taking of food, what is the capability of taking the food of Chinese are very different from the others. So when we are going through this observation, we see that the customs and all the characteristic feature of the people entire the world is play a critical role. The next question is the climate change, dust into the atmosphere. Climate change is a very big problem for our good Mother Earth. because it's probably not going to kill all the species into its own, because a lot of different types of hazardous activities has been done by the people in these developing countries, also play a very critical role here. And the next is, but as the nutrients rich fall out from the disaster land in the water, it becomes food for algae. And they exploit in population, releasing chemicals that can act as anything from a skin irritant to potent neurotoxin. And that's the very dangerous toxins that can also play a role in the extinction of the masses. Plants or lands can pick up the compounds in their root and pass them on to herbivorous animals. Algae growth is favored by the warmer temperature. So you can say that it's also play a role in global warming. You get accelerated metabolism and reproduction of these organisms. And the effect appear to be enhanced for a species of toxin-producing cyanobacteria. This is the symbol of mass extinction. What this symbol actually says, it's the symbol shows our Earth is surrounded. When you are going to do any unethical work on Earth, then what happened? It will extinct. All the five already we know that extinction happens. So it may be possible that the next extinction will be done on the basis of unethical behavior of the human being. We are selfish, so it's a symbol shows how the overall features that human beings are doing, all the work doing on the earth is unethical. So beyond this, there is a lot of characteristic feature of algae is too. How algae play an important role in our economic development of the world, economic development of the nation. When we are talking about the significant role, the different types of algae, that is blue-green algae, green algae, red algae, and brown algae. But the important feature is we are using a lot of plastic, and it causes very important and very hazardous problems in our society, in our environment. So to reduce the level of carbon dioxide, so we have to be use algae as an important considered in the different field, from science, for the use of science and technology. You use it in the field of carrying bag making, as well as the making of the cover of the mobile phone, as well in the field of food, and also into the chemical. There are a lot of chemical are present in algae, so used in different fields of in our everyday life. So the first thing, the brood-green algae, they are also known as cyanobacteria. They appear blue-green color and contain chlorophyll A, B, and phycobilins. There are a lot of economic importance. But now is what is eutrophication? These eukaryotic marine organisms have no root, flower, and a stem. It plays an important role in alkaline reclaiming, which is used as a soil binding agent. They are economically important in a variety of ways, which are discussed below. In the field of food, algae are healthy source of carbohydrates, fats, proteins, and vitamin A and B, C, and E, as well as in the mineral like iron, potassium, magnesium, calcium, manganese, and the zinc. Into the present aspect, when you are going to comparison with him, we can say that in this pandemic, the most focus of the people are on the nutrients, how they improve on immunizing capability, how they immunize. So all these materials that chemical or nutrients present in the algae can improve on health. Hence, people of the countries like Ireland, Scotland, Sweden, Norway, North and South America, France, Germany, Japan, and China use it as a food ingredient. That's very important in this aspect. And next is the fodder. Algae are also used as a fodder to feed livestock such as cattle and chickens. That also will help into the improving the digestive capability, as well as the what is the important ingredient required to the cattle. And so it relies on grain and different nations, including the North European nation of Denmark, Sweden, and Norway, etc. Like this pisciculture, the fish farming, algae play a very important role because it helps into production process. Fish used plankton and zooplankton as food. It helps in maintaining the health of marine ecosystem because algae are naturally absorbent of carbon dioxide and also provide oxygen to the water. But a lot of other algae like cyanobacteria, that's when we are discussing in another sense, it's play another role. Fertilizers. Algae are rich in minerals and vitamins, so they also use a liquid fertilizers which help in the repairing of the level of nitrogen present in the soil. Reclaiming alkaline blue-green algae help into the reduction of the high concentration of alkalinity in the soil. And the binding agents are algae act as a binding agent against natural process such as erosion. Biological indicator, algae also we have as a biological indicator because it is very sensitive. If there is a slight change in the environment, their pigment change or might get die. Water pollution is checked with the help of algae like euglena and chlorella. The above all the discussion related to especially into the mass extinctions are with a special reference to the hypothesis of the role of toxin producing algae in the phenologic mass extinction based on the evidence from the geological record and modern environment. Et al. Castle. Thank you, sir. Yes, sir. Yes, sir. Thank you.
Mr. Mani, Dr. Manish Kumar. Please. Respected sir, honorable teachers and dear colleagues and aspirants, today I say about algae and its beneficial use. I request everyone to listen carefully. A Swedish botanist Linnaeus 1754 was the first to use the name algae for a particular group of plants.
The word algae, singular alga, is derived from a Latin word alga, means seaweeds. The science that deals with their study is called algology. All the members of algae are autotrophic except Cephalurus, which is present on tree leaf. The study of algae is known as phycology. Everyone knows. Laymen have variously named algae as cornscams, frog spittles, water moss, and seaweeds. Algae from the various phyla show striking differences of color, and these often offer a quick guide to a preliminary classification of an alga. The algae as a group have now no official existence. It is a collective term for all those chlorophyll-bearing organisms which are alveolate. Many of them are small and unattractive. However, they are important members of the plant world, and several of them are significant to man in many ways. The growth and development of an alga proceed through a number of distinct morphological and subtle cell stages. It is widely accepted hypothesis that each phylum or division of alga originated from a unicellular ancestors. Many features indicate that each group is phylogenetically related to others. The vegetative structure of algae shows a wide variety, and it ranges in form from unicellular to complex multicellular phyla. Thallus is a structure which shows no differentiation into root, stem, and leaves. The algae can afford this simplicity because, with only a few exceptions, they are water dwellers. Even they are active only in very moist places or in times of moisture. Sexual reproduction algae in general pass through two phases of development to complete their life cycle. These are the spore producing phase, that is the sporophyte, the gamete producing phase, the gametophyte. Most of them are among the simplest in the plant kingdom. In all, there are about 30,000 species which are allowed in this world. Some important algae are Ulothrix, Spirodera, and Oscillatoria. There is a presence of chlorophyll and associated pigments in algae, mostly aquatic, but some are terrestrial on moist places and some are endophytic also. Plant body may be unicellular or multicellular. Algination of generation is not well marked in the life cycle. The process of algination of generation was discovered by Hofmeister in 1851. Humans derive their nutrition from algae. They have also begun to use alga as food. Porifera is most important red alga used as human food. Monostroma is used as common food only in Japan. Alga are often mixed with rice and fish and consumed as salads, soups, etc. The adult forms are called limu in Hawaii, sao in China, and lamu in Tahiti. Chlorella has been used as flour for making crackers and biscuits on experimental basis. Nostoc commune is consumed as food and is called uduk. Human in this society has transformed from the primitive hunter-gatherer to the present-day technologically dependent society. Initially, humans domesticated certain alga to fulfill their needs. Humans themselves are as a unique resource for the development of a nation, provided they have some physical and psychological help due to algae. There has been a rapid advancement in the diagnostic tools, surgical and other approaches, including immunotherapy, hormone therapy, and gene therapy to treat human ailment, that is disease. Different kinds of ointments, pills, and other pharmaceutical products are made from other. Many commercial products are extracted from algae and their cell walls. Alginates are also used in food industry for filling creams, thickening, soaps, sauces in domestic industries as dispelling agents in ointments, creams, toothpaste, shampoos, etc. The major components of agar is agarose. The material is prepared by washing, shorting, and boiling or autoclaving. Because of the presence of phosphorus, potassium, and some trace elements, the seaweeds in many coastal regions of the world are used as fertilizer. They are either mixed with some other organic materials or are allowed to rot in the field, as for example, fucus. Besides this, algae are also used as antibiotics, diatomite, and land reclamation. Conclusions of this topic is this: it is extremely difficult to define the group algae as they exhibit enormous range in vegetative structure, reproduction, and life history. The study of algae, number of plants belonging to this group baffles our imagination, and it becomes
extremely difficult to draw any concrete conclusion. It performs all the vital functions of life. There are enormous variations in the life cycle of various algae, and they present difficult in drawing any safe conclusion. Thank you, everyone.
Okay. First semester. First semester. Now Vikas Kumar from MSc first semester. The topic of your paper is economic importance of algae. Vikas Kumar, please come.
Good morning, my dear. Good morning, my honorable guests, teachers, and my colleagues. And this is my glorious day of my PG department. Today, this is conduct for important this e-seminar on a very honorable topic. My topic is economical value of algae. So, algae have many importance in our day-to-day life. Algae has as a food, as a source of mineral, as manure, as medicine, as nitrogen fixation, industry, as a food. So algae provide many ecological importance as well as many food importance in our daily life. Algae as a food, just like carbohydrate, protein, vitamins, many inorganic and organic compounds form an important ingredients of some algae. When these ingredients are present in large amount, huge constituents of our food, just like chlorophyll. Many classes of algae, just like chlorophyll, alga, form be used as vegetables after drying and salting. Chlorella contain a large amount of protein and fats, including vitamin A and vitamin D. It also form a very suitable food for a space flight. Just I know, vitamin D is a good source for provide for alga and many diseases just like Leukosolamia and many diseases like scurvy are caused with deficiency of vitamin D. So just I think about that. Just I prepare a medicine on a source of alga, just like they have constituents of vitamin D that solve the problems of Leukosolamia. Just another classes just like Phyophyceae, Limeria sardacea have got broad food value due to presence of protein, fats, and carbohydrates. They are also rich in iodine and therefore protect us from guanine disease. Rhodophyceae, just like ice mosses and Porphyra, provide with vitamin C and vitamin B12. Cyanophyceae, just like mascot, spirulina, taken as food in China, Java due to presence of protein as a source of mineral. Laminaria. Phyophyceae and Polyptyphonia rhodophyceae are the prime source of iodine and bromine respectively. As manure, starvation, marcosis are used as an inorganic manure for a standing crop in farm costs. As medicine, chlorella is as antibiotics called chlorella, which is very useful against disease by gram-positive and gram-negative bacteria. Nitrogen fixation. Blue-green algae like nostoc, anabena, cyanobacteria fix elemental nitrogen and thus increase the soil fertility. These algae are confined with the presence of heterocysts. This is a special cell present within a nostoc in the presence of trichome, which form a site of executing nitrogen fixation in a standing paddy crop field. According to PKD 1939, in industry, agar starvation LG and diatoms are the major industrial products. Agar are often from member of Rhodophyceae, collectively called Acrophyceae, which include Gelarium. Agar is provided or jelly-like substance used for bacteria culture in laboratory. It is used in the preparation of medicine. It is also used in baking and confinement industry as emulsifying agent. In cosmetics, textile, leather, and paper industry. It is also used in legislative during a cosmopolitan. Starvation another is cell wall derivatives of contrast crop that is Rhodophyceae class, which is used as an emulsifying and stabilizing agent in ice cream, chocolates, and toothpaste. Algae is also have a cell wall derived from fucus, which is used in preventing formation of crystal in the ice cream. It is also used in rubber, tire, and paint industries. It stops bleeding from part of our body. It also uses preparation of soap, cream, and soap clothes. And another is algae used as fuel, like coal, petroleum, and gases are all their energy to photosynthesis in ancient algal plants. Unlike coal, they are laid down in land on whether well or gases were generated in marine environment. Algae is useful to our day-to-day life, or our everyday life depends upon the uses of algae. So, with the help of algae, with the familiar of algae, with the friends of algae, I solve, I depend, I use each and everything. So, this is more useful for our day-to-day life. This is my glorious day of our PG department botany. So thanks for our principal sir to organize this type of program for me, and also my vice chancellor sir, vice chancellor sir. Thanks for all of you. Thank you, sir.
Thank you, Vikas. Now it is the lunch session, and at 3 p.m. we have very important lecture from Dr. Samit Ray, Department of Botany, Vishwabharati, Santiniketan, and at 4 p.m. another speaker, Professor David Kavule, delivered his important lecture. Both the lecture is on biofuel, so I request all the participants, all the persons to be at 3 p.m. Thank you. Lunch break.
Professor Kumar, this session is over. This session is over?
Next session is 3 p.m. sir. This session is over.
This session is over? Hello?
Yes, sir.
Can I speak to K.K. Sahu, who is referred he was reporting this session?
Yes, sir.
खत्म हो गया लगता है खत्म हो गया है देखो
हाँ सर,
खत्म हो गया है देखो
हेलो
सर लोग बाहर गए हैं सर।
हेलो
गणेश बोल रहे हैं।
हेलो
गणेश बोल
हाँ
Yes, sir.
गणेश
यस सर।
बोल रहा हूँ हो गया क्या लॉग आउट हो गए क्या बीएन वर्मा
कौन सर? हाँ सर पूछ रहे थे तो हम बोले सर अभी रिपोर्ट कर देंगे सर आप हम कॉल ही करने वाले थे आपको सर।
अच्छा ठीक है चलो हाँ जी सर हम तो थे ही नहीं सर वहां पर हम अभी ऑफिस में है उसमें से आपका कमेंट जो है ना सर वो आप 2 लाइन लिख के भेज दीजिएगा उसमें हम प्रोसीडिंग में लिख देंगे
बोलिए हम आ गए। हेलो हेलो। Now is the time for technical session third, and in this section, the lecture of Professor Samit Rai, Vishwabharati, Tanti Niketan. So I just is Professor Samit Rai online?
Can you hear me?
Yes, yes. Good afternoon. Good afternoon, Professor Rai.
Yes, thank you.
So I would like to request Professor Samit Rai to deliver his lecture. Hello, sir.
The screen is, you can see the screen?
Yes, yes, the screen is clear and you are audible to us also.
Okay.
Okay,
Should
sir.
I start?
Okay, sir, start, please.
Okay. So, first of all, good afternoon to everybody. And I express my deep sense of gratitude to Ellen Mithila University administration in general, and in particular, the Department of Botany, Ellen Mithila University, for inviting me. And I feel honored that I get a chance to share some of my thoughts with the students, senior students, young faculty, and the faculty members of the department. I should also take this opportunity to thank Professor B.N. Verma Saab, who introduced me to this department. With Dr. Verma, my association is very long. It was early 90s that I used to visit B.R. Ambedkar Bihar University, Muzaffarpur. And when early morning I used to get down at station, Dr. Verma used to be over there to receive me. Such a senior person, it was a great gesture on his behalf. And for many occasions, refresher course lectures, PhD Vaivavosi, I have visited B.R. Ambedkar Bihar University. And Dr. Verma has also visited Vishwabharati to conduct Vaivavosi of my student, PhD student. So it is a long association, and it was so kind of him that he remembered me after such a long time, and that also for such an auspicious occasion. And for Ellen Mithila University, I have checked some PhD thesis because Professor Amarendra Kaur is a long-time associate of mine, though he is very senior to me. But we have a very cordial relation amongst us. And he has visited Vishwabharati several times for special paper examination, PhD thesis, Vaivavosi, and for many other occasions. So this is my close connection with these two universities. Now, at the very outset, I would like to tell everybody over here that the topic of my lecture, this biofuels from algae, prospects and problems, this is not a topic that I am technically qualified to give a lecture on, but my expertise in the field of morphotaxonomy, cytotaxonomy, phytoplankton diversity, ecology, scanning electron microscopy, and some biochemical works like nitrogen fixation and other things. I chose this topic from my teaching experience. I am teaching algae for last 34 years. And what I would like to bring to the notice of the participants that there are so many promises to keep with regard to scientific issues in general and scientific achievements related to algae in particular. And in this regard, I would like to thank Department of Botany, Ellen Mithila University, that they have chosen algae and human affairs as the focal theme for this webinar. So I will discuss some points related to the biofuels from algae, and I will also request the audience to think over the issues that I raise. Those who really want to progress in the field of higher studies, those who want to take up scientific research as their career, want to go ahead with algae or plants in general, they should keep certain things in mind. So I begin with my first slide, that is bioenergy. Bioenergy, everybody is aware of bioenergy, though the source of energy is definitely the sun, but in several forms that we utilize that energy. In several forms that energy is stored in nature, and we, the human society, with our great knowledge of science, how to exploit things, we exploit that source of energy. So bioenergy is the renewable energy created from material obtained from natural resources. And natural resources, all you know, is a vast thing. Bioenergy is use of any organic material such as forest thinning, residues, agricultural wastes to generate heating and electricity. We know that within biomass is the organic matter which has stored sunlight in the form of chemical energy. So the photosynthetic products in various forms are stored in the as organic matter, and within which remains the chemical energy. Biopower or biomass power is the use of biomass to generate electricity. 6 major types of biopower system I will just briefly describe here. That is direct fired systems, that is you take the material, the feedstock, and directly burn it to produce steam, and from steam captured in turbine you run the generator, then convert it into electricity. Co-firing system, that means if you have coal-fired power plants, you can take the feedstock along with the coal, and the co-firing that reduces the production of the sulfur dioxide emission, and supplementary energy source is thus used, and it is highly efficient. Then there is gasification system, use of high temperature and an oxygen-starved environment that converts biomass into gases, and that gas can be used as a source of energy. Then anaerobic digestion to produce methane, and then there is methane, use of methane, everybody is very well aware of. Most facilities burn methane in a boiler to produce steam for electricity generation and for industrial processes. Then there is pyrolysis, that in addition to gas, liquid fuels can be produced. That is when the feedstock biomass is heated in absence of oxygen, and the biomass turns into a liquid called pyrolysis oil that can be used for as an energy source. Right. Then why people use this biomass? Because we also know that people who thrive surrounding the forest or the village people, they always collect biomass in various forms, particularly dried plant products, and they use that for firing and for various other purposes. So it is renewable. This is the most important thing, and that is the most challenging point with regard to the fossil fuel, that once that is burned, it is not renewable, though there are various theories regarding the formation of the fossil fuel. But in case of this bioenergy, what is readily available, the sources of biomass such as crops, manure, and garbage. As you convert this year's crop to fuel, you are growing another one for next year's fuel. So one side of the forest you cut, forest or the afforestation process, and on the other side you plant trees so that the next year you get a fresh crop. Renewing takes just a long time as it takes to grow or gather, and that can be as little as months in some cases. There are very good fast-growing plants, particularly of the leguminaceae, which produce a lot of fuel. Then it is carbon neutral. Carbon neutral in the sense, it is not that after burning them, the carbon dioxide is not produced, but the carbon dioxide that is produced, that from the atmosphere, which is absorbed into plants during photosynthesis, and when the plant decay or is burned, that carbon goes back into the atmosphere. So that carbon dioxide will again be utilized by the next crop, the growing plant. So it has been shown that the amount of carbon that the biomass will release into the atmosphere and the amount that they extract from it. So there is a good balance, so it can be said as carbon neutral. So biomass fuel are considered to be cleaner as compared to the fossil fuel. Then it is cost effective, but there may be some debates on this, the cost effectiveness, how do you calculate it? I am not going into the details of that. That is a very different issue altogether. But for the village people, for other persons, it is cost effective. And it is abundant. Biomass is available in large quantities all over the world. Large pipelines across country, borders and oceans are therefore most likely not necessary. This is a huge advantage that you can grow this renewable biomass wherever you want to. There is one significant disadvantage, that is requires space. So whenever you plant trees, a lot of space is required, and sometimes it so happens that you utilize the space which is needed for growing the food crop. That is a very big challenge, that how can it be that this plant material, the renewable plant material, is grown for fuel, then what will be the fate of the crops that the farmers are supposed to grow? So that is one real issue. So with this, we come to a very significant issue, that what made us think of alternative sources? What made us think of alternative sources? First thing is that we are all aware of that there is energy crisis. Finite nature of ubiquitously used fossil fuels. This is being discussed maybe for the last 100 years, that what will happen when this fossil fuel reserve will be exhausted, whether it is crude petroleum, whether it is gas, or whether it is the coal. And parallelly, the world population growth is reaching an estimated 11 billion at the end of the century expected, and global energy demand is expected to increase by 37% by 2040. We are in 2021, so another 19 years, and 30% increase in the global energy demand, because with more we are getting accustomed to the modern life, fast growing life, very sophisticated life, with the use of energy in the form of electricity, in the form of fossil fuel, are growing and growing and growing. The use of fossil fuels such as petroleum, coal and natural gas accounts for 85% of the global energy consumption. That has raised major environmental concerns. So it is not only that we are dependent 85% of our global energy consumption on fossil fuel, but it is harming our environment to a great extent, particularly the release of the greenhouse gases. But there is another huge problem, that is the geopolitical concern. Uneven distribution of fossil fuels in the world, highlighted by the presence of 63% of global petroleum reserves in the Middle East, creates an economical imbalance and geopolitical concern. We are well aware of that when India is purchasing this crude from Iran, America says there will be economic sanctions on you if you are purchasing from Iran, but Iran is our greatest supplier. So the crude producing, petroleum producing countries, they are controlling the price of this crude, and our economy to a great extent is dependent on that. So this geopolitical concern is another issue which makes us think about an alternative source. Use of fossil fuel increases amount of greenhouse gas emissions, as I have told earlier, and significantly contribute to environmental damage, affecting global climate and ocean acidification. Not only that, this global climate change, which is reflected in various storms, tsunami, earthquakes, and other things, they affect agriculture, the source of the sustenance of this huge human society, resulting in diminished yields due to droughts, heavy precipitation, and high winds, and also in countries like India, the flood waters. So we are in a difficult situation with regard to the energy. So whatever the remedy we find, there is an fallout of that which makes human society suffer. Currently, a mere 13% of world's energy consumption is renewable. It is in the form of solar, it is in the form of nuclear power, and 10% is derived from biomass. While the development of multiple renewable energy sources, such as cellulosic ethanol, solar, wind, and nuclear, is needed to meet the future world energy demand, extraction of high-energy compounds from algal biomass is a promising alternative needing scientific research and development. So this is one point that scientists have gathered after long research, that how can the algal biomass be used. So algal biomass is a crude term to use the thing, but it is the biochemical nature of the algal cells, the biochemical potentialities of the algal organisms, which is the promising alternative. So to get a good amount of lipids or oils from the algal cells, you need a good amount of algal biomass. So before I proceed further, let us discuss a little bit about algae. Frankly speaking, this algae and the human welfare is the focal theme. But before we proceed with very practical application of algae or the exploitation of the potentiality of the algal mass, we should have a very clear idea of what this group of organisms mean. We all know, the students of botany, whatever the level at which he or she is studying or teaching, algae is a big heterogeneous assemblage. Why should we be bothered about the heterogeneity of this assemblage? Simply because of the fact that if we look at the origin of the eukaryotic algae and its gradual evolution, the phylogenetic relationship, then it is clear to us that the algal organisms represent big genetic heterogeneity. Some organisms are just genetic chimera because of which has been explained through the theory of endosymbiotic origin of the chloroplast, mitochondria, and the origin of the eukaryotic, the polyphyletic origin of the eukaryotic algae to certain groups. Certain groups are, of course, monophyletic. So, how do we explain in short this heterogeneity? That prokaryote, they can be prokaryotic, eukaryotic, but here is the big question. Prokaryotic means the cyanobacteria, which are not algae anymore. Algae means the eukaryotic organism, and these cyanobacteria are placed in the Bunge's manual of systematic bacteriology, and they form of the various clades of the eubacteria. Cyanobacteria form a distinct clade in that, but it is very distinct from other algae, other bacteria, because they are having the chlorophylls, carotenoids, phytobiliproteins. capable of oxygenic photosynthesis due to coexistence of photosystem 1 and photosystem 2. And of course, there are certain metabolic pathways, particularly this oxygenic photosynthesis, which is very different. Algal members, they may be just one new and unicell to a seaweed, which is 200 feet, 60 meters long. Simple spherical unicell to highly differentiated thallus, and the genome may be represented by one circular chromosome to hundreds of rod-shaped chromosomes. Then comes a very significant part, that is the ultrastructure of the chloroplast and flagella, which have really shown us that how diverse this particular group, not only with regard to the chloroplast structure, the simple chloroplast, the complex chloroplast, and the various combination of pigments with the structure of the chloroplast, the metabolism that undergoes within chloroplast, that also varies, and that is the crux of the point that the scientists have studied quite well to get into this at the base of the problem of this biosphere. Chemical diversity with regard to pigment, reserve food. Here again we find two very significant aspects. That is the pigment, because pigment composition is at the basis on which the photosynthetic efficiency of the organisms are concerned with. What will be the efficiency, under what light condition, what will be the output, which pigments, those organisms which are showing chromatic adaptation, what kind of photosynthesis will be going on, and what will be the efficiency of that photosynthetic process. So pigment composition will have to be thoroughly studied, which is also reflected in the classification of algae. And then the reserve food. This is another issue which we should be well aware of, because the reserve food is one which will be gradually be converted into, which shows a lot of diversity, starting from cyanophycean starch, Floridian starch, laminarin, chlysolaminarin, fats, oils, everything has been are the product of the algal metabolism. Then comes the strict celibacy, the very complex life cycle. Life cycle process is important because at various stages of the life cycle, there are certain metabolic changes that take place. This diversity has not been formed suddenly. In the Precambrian, there were the cyanophycean members. Gradually, they became the most dominant part throughout the ecosystems throughout the world, and then there was eukaryotization, the eukaryotic algae at the early part of the Paleozoic, and then gradually various groups of algae originated. They originated firstly in the form of unicells, and then gradually there was multicellular formation, development of sexual reproduction, and gradually there was genetic diversity. So diversification of algae must be understood at the cellular, biochemical and molecular level, not only morphological. Basic difference with the higher plants lies here, that the morphological variation of the algal organism need not be reflected in the taxonomy. They are mostly done with the help of cellular, biochemical, molecular and ultrastructural parameters. So molecular phylogenetic studies established that the algal organisms may be distributed in several kingdoms. If the cyanobacteria is in bacteria, then chlorophyta, glaucophyta, rhodophyta in the kingdom Plantae, and the rest can be in chromista or chromalveolata, or some may be in the rhizaria. So this is, I give a glimpse of the diversity in algae, without which we are not in a position to exploit the potentialities of algae. So why algae is so important? Why? Because from the very beginning we know that it is in the very recent years, maybe after 1970s, that there is a great thrust on this biofuel aspect. But it is since the 18th century, since the time of Linnaeus, that scientists are very well aware of the potentialities of algae. Think of the cyanobacteria. that they first evolved the oxygen in the environment and their potentialities to fix nitrogen. So fixed carbon, fixed nitrogen added to the environment for the growth of the other organisms. So that was the beginning of aerobic metabolism. Then think of the later studies. Photosynthetic algae fix over 40% of the Earth's carbon. So this is a very significant observation, because without fixing carbon, we cannot proceed further, lest apart this biofuels. Then oceans cover 71% of the Earth's surface, and phytoplankton are the most abundant there, and also in freshwater bodies. Plankton are important to prevent global warming only through their rapid rate of photosynthesis. Prochlorococcus marinus, I don't know whether students are aware of this organism. This is a green cyanobacteria, that means cyanobacteria which have chlorophyll A and B. And certain populations of this particular genus, they produce phycoerythrin-like pigments. So they are sometimes referred to as the green cyanobacteria. But these Prochlorococcus marinus, which have been found in the 1980s, they are the most efficient photosynthetic organisms presently found on Earth. In oligotrophic condition, they contribute 30 to 80% of the photosynthesis. So playing a big role in carbon fixation and reducing the global warming. Then many of them are the primary colonizers. Then think of coral reef. Algae constitute 3/4 of the tissue within the living corals. Then the seaweeds. From early civilization, the maritime people, they were so dependent on the seaweeds as food, feed, and the source of very essential elements like iron, sodium, potassium, and also their tremendous role in the ecology. They are vital for the health and ecology of the rocky shores. Total area covered by seaweeds as big as Africa, and they form the habitat for the fauna. And then comes this diversified group provides food, feed, nutraceuticals, pharmaceuticals, vitamins, proteins, ecological balance, oils, and above all, what Professor Kesri has given a detailed account of in the morning, that is the how to be what algae are added to the glamour world, and in the form of cosmetics, in the form of design, in the form of essential nutraceuticals, the antioxidants, the anti-cancerous one, the protein supplement, the fat supplement, everything that he has explained so nicely. So these, in short, exemplifies what is the big potentiality of this heterogeneous group of organisms. Not all of them belong to the kingdom Plantae, many of them belong to the kingdom Promista or Alveolata or the Rhizaria. Then why algae for biofuel? This is a very interesting subject, that when algae is serving so much for the human society, then why should we be dependent on algae for the biofuel? One point that I can just recapitulate from my earlier slides, that plant oils are a source of renewable energy according to a rapid increase in social demand for establishing a low-carbon society. So the biofuel sources, of which the plant oils are one, they are of low carbon potential, so which was needed for the to save our environment. Oil production for biofuels and biorefinery using higher plants and crops competed with food production and to increase those market prices. So if I am to grow soybean or jacropha or rapeseed to raise the plant oil, then naturally a lot of space will be taken, a lot of land will go, and that will go at the expense of the agricultural crops. So naturally the product prices will rise, and there may be some food crisis. Now, algae came into play, a new oil-producing organism, since algae do not compete with food production. Algae not only compete with food production, here the point is they do not compete with food production because algae is not for all the human population, they are a natural food alternative. They are used as food supplement or as nutrient supplement, but not the staple food for everybody, barring a very few instances. But that production of algae for any use for human benefit, that do not compete with the space which are used by the agricultural crops. According to their high productivity per unit area, prokaryotic photoautotropes such as cyanobacteria and eukaryotic microalgae were a suitable alternative. This the scientists gradually came to understand, because more and more sophisticated instrumentation, sophisticated processes were being developed, and whose application was being made in the field of phycology. The variability or the diversity of at the chemical, biochemical level were being revealed by the scientists, not only phycologists, but others also who were working with algae as the material that was being revealed, published, and accepted by the society. Like the example that Jayaprakash gave in the morning of the spirulina, of the hematococcus, of the cyanobacteria, Aphanizomen and Clostridium, all these. As many algae were found to grow rapidly and produce substantial amount of triacyl glycerols, oleaginous algae, it has long been postulated that they could be employed to produce oil and other lipids for biofuels. Microalgae are efficient in converting solar energy and sequestering carbon dioxide into storage lipids, with several species have higher biomass production rate and higher percentage of oil than terrestrial plants. So on the one hand, we have just discussed that the pigment composition of the algal cells, and they have the potentiality to change the pigment composition in the cell depending on the available nature of the light, incident light, so that they can change their photosynthetic process, photosynthetic potentiality. They have that flexibility, and on the other hand, that they part of their storage food they convert into the oil, which is the compound that is to be exploited. Microalgae predicted to produce 10 times more biodiesel per unit area of land compared to terrestrial oleaginous crops. It is at one study it is estimated that if the crops are to be planted, it may need 54,000 acres, but for the same amount of diesel, maybe if we do the algal mass production, maybe 6 acres of land may do that job. Okay. Then this is some photographs of the very potent algal triacyl glycerol producers, the Nanochloropsis, the Chlorella. Then Botrycoccus browni is the hydrocarbon producer, and then Senedesmus is another very interesting green algae, which are very potent in this field. Then we come to the microalgae chlorophytes. Generally use starch as their primary carbon storage compound. Some species accumulate neutral lipids, mainly in the form of triacyl glycerols under environmental stress conditions such as nitrogen starvation. So focus on this particular point. First of all, that microalgae, those which are unicellular or maybe colonial, and mainly those from the green algae, the chlorophytes, they produce starch as the reserve food, and the starch is produced within the chloroplast. This point must be very carefully understood. The chloroplast produce a starch, and starch is produced within the chloroplast. That is the reserve food for green algae. And some species accumulate neutral lipids. Now, why the lipids are formed in the cell? They are also storage products. And why under starving condition? Because the storage products, they are needed for the stress conditions, because when they are only congenial environment, when there are this stressful condition, when they cannot have their food readily available, then they can utilize that stored matter for their survival and to carry on with the metabolic activity. So when the congenial environment returns, then there is they utilize that stored matter to revive their metabolic activity. In response to environmental stress, they reduce proliferation and start producing triacylglycerol tag. Environmental stress, they reduce proliferation. It is also the origin of, if we look at the origin of the sexual reproduction, then also we find that as they find the situation growing very harsh, they go for sexual reproduction, produce a structure of a hard wall like the zygospore, oospore, so that to thrive in the harsh conditions, and they germinate when the congenial environmental conditions return. And as a result, the cells will have to accumulate a lot of storage material, and in this case, and for our interest, that is triacylglycerol, which we will exploit. Of the three biomolecules formed, carbohydrate, lipid, proteins, three important components, lipids have the highest energy content, which make them high-value molecules. This is a standard thing we all know. Harvested lipids typically undergo transesterification with methanol to be converted into fatty acid methyl ester that can be used as a transportation fuel in the form of biodiesel. But that is theoretically possible, and some experimentations have been done. But whether that will be economically viable, that point I will come later. The remaining biomass may be converted into methane gas, or the carbohydrates may be transformed into ethanol by dark fermentation. This is another very significant point, simply because of the fact that only one mechanism and one product that may not be economically viable. So once we take out the triacylglycerol for the esterification and formation of the biodiesel, then what we'll do with the rest, we can produce it for use the biomass for methane. Then carbohydrate may be transformed into ethanol by dark fermentation. That is a very, very practical approach. Other factors that attracted attention of microalgae are some species' ability to rapidly grow on open pond or closed systems or land otherwise unsuitable for farming. Before I describe this point, one point that I should highlight, that is when the algae are growing very luxuriantly under very favorable environmental condition, they are not producing much amount of lipid because that is not needed in the cell. But when the algae finds the stress conditions, they will not grow luxuriantly. The biomass production will be less, but there will be a lot of lipid production. So they are mutually exclusive. So that is a big problem with the scientists, because if you want to sustain it economically, then with the luxuriant growth there must be a lot of lipid production, so that there will be huge biomass, there will be huge lipid extraction for the biodiesel and the rest of the biomass for other things. So that is one thing which had to be overcome. Other factors that attracted attention of microalgae are some species' ability to rapidly grow on open pond or closed systems on land, otherwise unsuitable for farming. So this is one advantage, that there won't be any clash of interest with regard to the space. Thus, not competing for arable land like oilseed crops, their potential for year-round cultivation, growth in saline water. Saline water growth is another stress condition. Recycled municipal wastewater and landfill leachates, co-production of high-value compounds along with biofuels, and most importantly, their ability to accumulate impressive amounts of lipids under some conditions. Several studies have shown that lipid content of 20 to 50% per cell dry weight are commonly observed in reaching up to 80% cell dry weight in some species under certain stress conditions. So challenge is, the point is that we are armed with the finding that under stress condition they will produce a lot of lipids, but they will not grow luxuriantly. So we will have to find other ways. Microalgae generally use starch as their primary carbon. This is the pathway. So what happens? That carbon dioxide is taken, and then through Kelvin cycle, this 3-PGA, phosphoglyceric acid, is produced, the pyruvate. And then at this stage, the bicarbonate ion is taken by acyl CoA carboxylase. That creates the malonyl coenzyme A, and ultimately that enters the fatty acid synthesis cycle, and then it goes to form free fatty acid. That fatty acid comes out of the plastid, and then it takes up coenzyme A and forms the acyl coenzyme A, and then there is glycerol 3-phosphate from DHAP, dihydroxyacetone phosphate, to glycerol 3-phosphate dihydrogen, and then the glycerol 3-phosphate ultimately comes over here. It combines with the acyl-CoA, and through several processes, it forms the triacylglycerol, the lipid. That is the neutral lipid. Because within the plants, we know within algae, there is the formation of neutral lipids and the polar lipids. Neutral lipids and the polar lipids. And the polar lipids are those like the phospholipids, glycolipids. They are the forms which are used for the formation of the membrane, but the neutral lipids like the triacylglycerol, they will be within the membranous structure, and that will be the storage one. Okay. So this is the pathway, and the major lipid components in algal organisms, as I told, the diversity of algal lipids and its ability to modify according to environment made algae ubiquitous. So we are targeting algae as a source material, but what algae is doing for itself is to remain alive under difficult situations, and as they are potent to change their metabolic pathways, to change the ultimate product of the metabolism, they are thriving throughout the world. The lipids may include polar lipids, neutral lipids, hydrocarbons, etc. Neutral lipids basically consist of triacylglycerol, which we are targeting, and polar lipids include sphingolipids, glycolipids, phospholipids, and sterols. So, certain algal species produce long-chain or highly unsaturated fatty acids that are unique to those taxa, because it is the fatty acids which will be ultimately forming the lipid. Variations in the component of fatty acid of algal oils can be observed in response to growth conditions. In case of microalgae, the oil accumulating cells are themselves fully photosynthetic. Now, here I just give one food for thought, that whenever we think of exploiting certain organism, we generally think of genetic engineering, and or rather we think of biotechnology. And a general impression of most of the students generally that biotechnology means genetic engineering. But unfortunately it is not so. Biotechnology is application of some technology to exploit the biological resources optimally. We can change the growing condition of an algae, and we can change the final product produced by that organism. We need not go for any tinkering of gene, that is the genetic engineering. In some cases, genetic engineering is needed. We will see it here also, that where certain particular genes are to be silenced if it is kept in its mother machinery, or it can be cleaved out and put into another system for the production of that particular protein or that particular lipid. So, conspicuous formation of lipids by microalgae is the result of accumulation of storage triacylglycerols. As the algae continue to photosynthesize under conditions that inhibit cell division, massive quantities of oil can be produced and can occupy the majority of cell. So here again that point that if the cell division is inhibited, then only the lipids will be produced. But we want things to go in a different way. Triacylglycerol production in most algae is correlated to severe stress condition. So see, here we are again coming to that heterogeneity of algae and the flexibility of the system, that under stress conditions for various, it may be salinity, it may be pH, it may be temperature, it may be different delimiting chemicals, that the nature of the metabolism will change. That scientists have studied so thoroughly. The stress conditions under which lipid production increases documented are nitrogen deprivation, sulfate limitation, phosphate limitation, desiccation, high carbon dioxide concentration also was observed to promote lipid synthesis. So these are so many factors. So where are we going to apply these conditions, these variable conditions? We will have to produce algal biomass under controlled conditions. It may be a raceway pond, it may be an open pond, it may be still further defined condition, that is the photobioreactor, various kinds of photobioreactor. Then there is production of hydrocarbon. We are all aware of this Botryococcus brownie, C17 to C34 hydrocarbons are found. So now we come to what are the steps in biodiesel production. Parent oil used in making biodiesel consists of triacylglycerol, in which 3 fatty acid molecules are esterified with a molecule of glycerol. In making biodiesel, triacylglycerols are reacted with methanol in a reaction known as trans-esterification. Methanol is used as it is least expensive. Trans-esterification produces methyl esters of fatty acids that are biodiesel, and byproduct is the glycerol. Alkalis such as sodium and potassium hydroxide are commonly used as commercial catalysts at a concentration of about 1% by weight of oil. Lipase enzymes are also used, but that will be expensive. Biodiesel is recovered by repeated washing with water to remove glycerol and methanol. So this is the crux of the mechanism. Microalgae biofuels suffer from high capital and operating costs due to low yields and costly extraction methods. Microalgae grown under optimal growth conditions produce large amounts of biomass but with low neutral lipid content. With microalgae grown in nutrient-starved conditions accumulate high levels of neutral lipids but are slow growing. Producing lipids while maintaining high growth rate is vital for fuel production because biomass productivity increases yield per harvest volume while high lipid content decreases the cost of extraction per unit product. So there is need for metabolic engineering, and here comes the real issue. So what is metabolic engineering? It is the practice of optimizing genetic and regulatory processes within cells to increase the cell production of a certain substance. For our case, it is triacylglycerol, of course. First, to pinpoint part of network that constrains the production. So, which is the part of the pathway which becomes the constraint? Constraint, that means that there is luxuriant growth, less of lipid. Lipid is produced to tide over difficult situation. So if the difficult situation is not there, if there is no stress, why should algae produce triacylglycerols, the lipids? It has no purpose because everything in nature takes place for a definite purpose. So that point scientists had to work out, and genetic engineering is used to modify the network in order to relieve these constraints, So then there is use of mathematical modeling to calculate the yield. So what kind of modification will lead to purpose, that for that mathematical modeling is needed. So there were several issues that were found, several steps that were found. One is the overexpression of the enzyme acetyl-CoA carboxylase, 3-ketoacyl carrier protein synthase of much use. But monitoring the activity of these enzymes didn't yield much. Then more successful was attempt to overexpress glycerol 3-phosphate dehydrogenase enzyme involved in supplying glycerol 3-phosphate required for tag formation. You just go back to, you just recapitulate that from dihydroxyacetone acetone phosphate, when the glycerol 3-phosphate was being formed, and the glycerol 3-phosphate dehydrogenase enzyme, monitoring that was required for more tag formation. Diacylglycerol acyl transferase enzyme catalyzing the final step of tag synthesis. At that point also scientists worked. Increasing the activity of acyl ACP, acyl carrier protein esterases appears to be a good strategy to promote continuous production of fatty acids and channeling them to strong lipids, storage lipids rather than membrane lipids. 72% increase was possible. Now the point is that the nature of or the efficiency of the stored lipid will depend on the fatty acid chain, and this enzyme, this acyl ACP, acyl carrier protein thioesterases, this enzyme is responsible for the length of the chain length of the fatty acid formation. So if we can want to achieve an ideal fatty acid length, then we should form a transgenic overexpression of thioesterases that can be used to change fatty acid chain length. So it will be compared that what was the chain length in case of the fossil fuel and what is the condition over here, and on the basis of that, some genetic engineering can be done for the change. Okay. Then comes another approach was to decrease lipid catabolism. That was another fantastic thought by the scientists that genes involved in beta oxidation of fatty acid as well as the activation of free fatty acids could be targeted for deletion by gene knockout methodology. Enzyme beta oxidation in microalgae, which are responsible for the breakdown of fatty acids in the mitochondria or peroxisomes, could be promising targets for microRNA silencing. So you think at what level scientists are thinking that unless and until you have a good knowledge and good background of the molecular biology, latest technologies of the genetic modification, genetic editing, and gene knockout methodology, microRNA silencing, all these are being used for this purpose. Then comes, these are the photographs of your photobioreactor, the open pond system, and then we will come to discuss. There is another figure that is Pseudochlorococcus uses starch as primary storage product, and 2% lipids can be increased under nitrogen depletion condition. Microalgae that are capable of heterotrophic growth on sugars were able to enhance lipid accumulation by 900% compared to phototrophic species. So if you are growing algae in under controlled condition, if you are adding additional carbon sources, fixed carbon sources, apart from the photosynthetic filtration of carbon, then there can be increase. But whether that will be economically viable, that is a big question. Chlorella were able to achieve 79.5% total lipids when fed with glucose and grown in darkness. So these are some of the experimental things which have come into being, which have been found by the scientists to augment our production of triacylglycerol, to exploit the mechanism of the algal cell, the metabolic mechanism and the genetic apparatus, to use it to its fullest optimization that they are trying. But what is in store for us? That is, can algae be economically cultivated and commercially scaled to make a material contribution to humanity's liquid fuel need? Whatever I study, that is the answer so far is no. Predicting cost varies from $0.42 to $70 per liter. So this will have to be compared with the fossil fuel. And another thing is that since the very beginning of the exploitation of this algal biofuel, which started in 1942, microalgae may be grown as a source of lipid for fuel. It was 1942, and then there was World War II. And after the World War, there was a big thrust in USA, Japan, England, Israel to grow microalgae on a larger scale. And Chlorella pyrenoidosa was found to accumulate as much as 70% of its dry weight as lipids. Efforts to produce alternate transportation fuel subsided after World War II because there was steady flow of fossil fuel and there was not much increase in price. But from 1970s onward, during the period of oil embargo and surging of oil prices, production of liquid transportation fuel from algae again started. So whatever we try, there is a reason behind it. And The point is, the research program focused on the cultivation of microalgae in open outdoor ponds, systems which are low in cost but vulnerable to environmental disturbances like temperature swings and biological invasions. Algal strains were screened for properties like high productivity, lipid content, and thermal tolerance. So gradually what happened, that this cost thing was a big issue, and several data are available on net, but what are the factors taken into consideration for the calculation of the price? There remains the basic issue. Because if you are giving extra carbon source, then the price will come something other. If you are using artificial light, price will come to else. If you are dependent on sunlight, the price will come to ends. If you are depending on only for natural photosynthesis, price will come to something. So they varied from 0.42 to $70 per liter. Aerial productivity, provision of nutrients, energy, and waste usage are key challenges. Supply of carbon dioxide, recycling of nutrients, nitrogen, phosphate is very essential. So all these are hiking the cost. Careful balance between energy and water consumption must be maintained. There is a tremendous amount of water needed. So according to one estimate, that if the transportation fuel of European Union is to be replaced by that of biofuel, biodiesel, then a pond of the size of the whole country of Belgium is needed. So is that possible or feasible? So major problem is energy balance of lipid extraction, maintaining suitable growing condition in open ponds, immense volume of water requirement, carbon dioxide and fertilizer required to allow the algae to photosynthesize fast enough to large scale. But still people are trying. How people are trying? That one main problem in lipid component extraction is to remove all moisture from algae. Dry powder is needed for lipid separation, but this requires more energy to power the process than the resulting fuel provides. Now, the engineers have given a new method. They are pushing jets of the algae, algal culture, and that of the mixing of the chemical which is removing the fuel from it, the fossil fuel from biofuel from it, that precludes the need for energy intensive drying process. So drying cost may be separated. Improvement in strain characteristics, as I have seen highlighted, this metabolic engineering and genetic engineering characteristics and cultivation method, estimated cost should be $300 per dry ton. Combined algal processing, plant capable of simultaneously producing algal biofuel, surfactant, polyurethane, and plastic composite. If from a single plant, so many products can be made. Expectation 10,000 barrels of algal biofuel a day by 2025. But here I want to mention that many companies were floated having billions of dollars from the private market were invested, but the present situation is not very positive. It is rather gloomy. Genetic modification of nanochloropsis, the marine unicellular form, has resulted in the doubling of lipid content in algae from 20 to 40%.
Professor,
yes, last line.
Okay.
Process economics enhanced by integrating biodiesel production into biorefinery producing a suite of products. So this is the scenario. So what I would like for particularly the senior students, you think of the future, definitely, by the exploitation or maneuvering of the biological system that is available. It can be anything. It can be algae, it can be fungi, it can be anything, it can be angiosperms. But so many issues are to be taken into consideration. But one, the silver lining is that when we studied all these parameters, we really come to know the intricacies of the biological system at its best. Thank you so much.
Thank you. Thank you, Professor Ray, for very informative and elaborative lecture on biofuel.
Yes.
And now the lecture is open for discussion. Any question from audience? Thank you, Professor Ray.
Okay.
For your nice lecture.
Yes. And I also thank you profusely for giving me a chance to share my thoughts.
No, sir. It's our pleasure.
And I hope people have liked this lecture, and maybe this gives them some food for thought.
No, you have given very informative lecture about, and our students will be benefited from this.
Okay. And if they like, I will forward this PPT to you for circulation.
Yes. Okay. Thank you, sir.
Yeah.
So kind of you.
Okay. And definitely I will give my written submission in another 5-7 days. Yeah.
Okay, sir. Thank you.
Okay. Thank you.
So now it is the time for another lecture of Professor B.B. Chogle. Is Professor Chogle online?
Yes. Hello, good evening.
I'd like to request Professor Chogle to deliver his lecture.
Sure.
Present now.
One moment, sir. 1 moment.
Entire screen.
Audible to you?
Yeah.
No, not this.
Oh, sorry. Hello.
Yes.
Okay. Are you able to see?
Yes. We are seeing and listening you.
Yeah. Screen is visible? Properly visible?
Yes, sir, properly.
Okay. Thank you, sir. Thank you. I was listening the last lecture. Unfortunately, I could not listen much. And I am afraid there could be some overlaps. But what I've done, there are problems with the biofuel, its economy, whether it is economically feasible, commercially viable or not. But we have no other alternative but to go for biofuels, and algae are the alternatives. Anyway, I have tried to orient my talk from the point of view of the students, botany students. What would be the role of the botany students in this endeavor? That is what would be my focus of talk. If we really go to, India's, our requirement for energy, any country, not only India, they are mostly depending upon fossil fuels for their energy requirements. And looking into the India, the situation is very different. Because as you know, that there are various sources of fuels like crude oil, coal, electricity, hydro power, thermal, nuclear power, etc. But the energy source in the form of liquid is very important because it is required for transport, for agriculture, and for other purposes. We do not have any other alternative. There are vehicles which are battery driven, but again, there is a limitations where high loaded vehicles, lorries or aeroplanes, trains, they require a diesel. Those can't be run on batteries. And therefore, you have to think of oil in a liquid form. Now, if we really see the Indian scenario, we are depending upon the foreign resource because our fossil fuel resources are very, very limited, and almost our 75% of the fuel, crude oil, comes from the other countries, where we have to waste huge amount of money into that. And this crude oil consumption, as everyone knows, is increasing every year. And it is estimated that by 2030, now our dependence on imported crude oil would be as high as 95% from the present 75%. Besides, see, it's a costly affair as far as our country is concerned. Not only it is an imported one, but also there are higher taxes also into that. So looking into all this, and more than that, what everyone has to remember, that there's an estimation, is a paper published by in 2009 by Australian scientists. It's based upon the statistic available amount of the fossil resources. And if we really look into world's consumption of fossil fuels of the year 2006, the reserve food of oil, gas, and coal will last further to 40, 70, and 200 years respectively. That means the crude oil will start depleting from the year 2046. So sooner or later, oil, liquid form of oil, is going to be exhausted. And we have no other alternative but to look for other resources. Now, besides shortage, cost, there are other aspects also we must remember, that because of the pollution, I mean the oil that is used, we have it's not only from the vehicles but also from the industries, CO2 emissions. And countries, here is a map where we can see the emission ratios. Although we are at base, but other countries are much higher, but we are also competing with others in that respect. Now, this rise in a CO2 has led to increase in a temperature, greenhouse effect, as we all know, that's responsible again for environmental degradation. So, I mean, as previous speaker told, that efforts are in progress, and now we are in a third generation attempt. The first generation was on biofuel production, was on a sugar cane, corn, or corn. Is on a food grains that are used and to produce ethanol. But again here, the problem is whether you will think of food production or fuel production. Priority certainly will go for food production than the fuel production. And therefore, again, people have started for a second generation type of biofuel production, and where cellulosic fuels, crop-based biodiesel production was attempted, because this was not involving the food grains. But again, here the problem is, for this you require again a land for this. And again, I mean, to meet the requirement, it is estimated that almost 20% of the forest land need to be deforested, which is not possible. So land again is again the problem is whether whatever the available land that is available present, whether you'll use for crop production or biofuel production. And therefore, it is now we are in a third generation where algae are considered as the source of biodiesel, because there's no competition with the food as well as there's no competition with the land. Again, there are certain advantages with algae, I mean the oil production from algae, and the food crops as well as the land area that is required compared to corn, soybean, palm, jatropha, coconut, which are used for biodiesel production. The oil yield of algae is much, much, much higher. And compared to the land that is required, other crops, is much less in case of algae. Besides this, there are many other advantages with algae, that algae can be grown on a wastewater, so where additional nutrients would not be required. And in addition to this, it will help to sequester the carbon because it's a pollution free, and that will help to reduce the levels of the carbon dioxide. Previous speaker also talked about the economics, and therefore the people are trying to hunt for value-added products like biopolymers, proteins, polysaccharides, pigments, amino acids, fertilizers, hydrogen, etc., so that the cost of production would be decreased. And also, algae can be grown all year around, unlike the other crops, depending upon the location. Because in India, for example, there are locations where you have a very few rainy days, and for most of the year, sunlight is available. So such areas can be used for open pond cultivation, while in other areas, cultivation can be done in a closed bioreactors. With this big background, now I would like to talk, keeping into mind the interest, what is the role of the botanist in this, particularly those who are involved in the study of algae. Because we know that algae only possible source. One second. And the question is now, which algae you would use, where you find this, and how to go ahead. Because normally algae grow in a mixture, and for any cultivation, you require many algae populations. It's said, this is based upon my own experience also, that native strains are always ideal strains. Because those are already acclimatized strains. For example, if you want to have a pond, say in Bihar, so look for a strain which is already available in Bihar. If somebody wants to have his cultivation system, say in Maharashtra, then he must look for a strain from Maharashtra. And again, in any state, you have many different zones. Like Rajasthan, for example, you have a desert where algae could be grown. And if you could isolate the alga, try to use the alga already growing into that area, would be the ideal situation. Now, the first phase in this requires, therefore, it's the collection of alga from various where alga grows. I mean, people probably would be aware of that, so maybe aware of that. See, there are sources like fresh water, brackish water, marine water, and within the fresh water, you can collect algae from standing waters like pool, puddles, ponds, lakes, impoundments, running water, springs, streams, rivulets. The other sources, as far as India is concerned, a major source are the estuaries and water and the sea, there Arabian Sea, Bengal, Indian Ocean, where one can look not only the open sea regions but also tidal pools. So then, in addition to this, in states like Uttar Pradesh, part of Bihar, some areas, Maharashtra, where because of the excessive irrigation and use of agricultural crops, soils have turned saline. There are many such saline soils which are waterlogged one. If you could isolate alga from this region, it would be the ideal strain to be grown into that particular area. Then the other places are hyper saline, like salt, because here algae grow at the stress conditions, and as a sort of adaptation, they have already inherited ability to produce more and more salt. I would also suggest to collect algae, to collect the soil sample from dry water body, garden soil, forest, deserts, seashore, estuaries, any soil, as well as tree trunks, leaves, milestones, rocks, and use those for isolation of algae. And we must also attempt to collect
हेलो हेलो डिस्कनेक्ट हो गया सर नहीं है मैसेज आ गया सर है ऑनलाइन
अच्छा, अभी 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 ऑडिबल हो गए? हेलो हेलो, ओके ओके ओके, रिपीट.
This one.
हाँ. Hello. Hello. Hello.
Hello. Yes, sir.
So I was explaining how to go ahead, how to isolate alga, and once you collect the material, then isolate. Either one can use a liquid culture or one can use a soil for isolating the strain. And as I told that, you need not worry if you don't have a culture room. You can use a north window where there will not be direct sunlight, there will not be heating. Then liquid alga which is cultured, collected in water can be used for isolation using agar sticks, agar plates, or agar tubes, and soil that are collected from dried ponds or forest areas or even open land, roadside, whatever the soil, either from deserts, from any area, that can also be used for isolation. What you have to do is you have to make a biophysical test where soil is placed in the bottom and medium is poured on the top. And then, or alternatively, in a petri plate, medium is taken and soil particles are sprinkled. Now, once you get alga, then you have to upscale it. Upscaling is essentially trying to get more and more biomass. For two reasons. One, a huge amount of biomass is required for oil production, but before that, you have to get the biomass in few grams to test some of the things, and this is done from agar plates to conical flasks of small volume, larger volumes, and then into bigger vessels. Now, the different tests that are carried are to find out how much oil that is present. And there's a qualitative test, and then the chemical extraction test, from which one can find out whether oil is present in enough quantity or not. Now, in addition to oil, one has to also try to find out how much protein, carbohydrate, pigments, and secondary metabolites are there, because those I will tell a little later on why there's a necessity to estimate protein and other products. Now, once you get alga isolated, you have to identify it, genus and species, using monographs, and then you have to conduct a couple of physiological experiments, because those are highly essential, particularly when somebody tries to grow into the field. Number one, to find out the specific growth rate, because you need alga which would be growing. Number one, the first that we have tried to find out, it is the liquid quantity is high, and profile also will tell you that the type of fatty acid that are required that are present or not. And then whether alga grows fast or not, that we have to find out. Then what would be the maximum density that alga would develop, form in a laboratory, because ultimately this data will be used for growing this alga in the field. Then also one has to test tolerance of the isolated strain to different environmental variables like temperatures, pH, salinity, oxygen levels, carbon dioxide. See, because this is highly essential, as you know that country like India, where we don't have a fixed seasons. Seasons are changing accordingly. The environmental temperatures would change. Because of the evaporation and the chemicals that are added, pH would change, salinity will change. Due to photosynthesis, oxygen levels would change. Then also one has to find out the nutrients that are required. And then simultaneously what is done is the for growing alga on a large scale, you cannot use laboratory scale chemicals because those are very costly one. And therefore the attempt is, for example, instead of nitrogen, inorganic nitrogen, any say sodium nitrogen, potassium nitrogen, it is urea is preferred because the cost is less. Same is true for potassium and other nutrients. And therefore one has to also test ability of alga to grow into low cost medium. Then one has to look for its culture constancy. That means whether the behavior of the alga is maintained for several generations, say in one season and then second season, third season, year first, year second, and so on. This is highly essential. Then resilience, it is the flexibility of the alga is very much, because with the say a country like us, the morning temperatures will be very low, afternoon temperatures will be high, and therefore alga should be able to adjust such fluctuating conditions. Then the next one is community stability, because in case of particularly open ponds are open one, and because of the wind, the soil particles would be added into the ponds, and which are the again sources of contaminants, including algae. So the alga that is used for cultivation should be able to compete the contaminating one. Then it should not be susceptible to predators. See, in open ponds, as I told you, that there will be grazers, there will be viruses, fungi, everything would be there, and alga should not be very susceptible. And all these parameters which I mentioned are tested in a small scale ponds, say of 500 liter capacity, then 1,000 capacity, 5,000 capacity, and slowly the volume is increased. Now, here is the oil content of some of the algae. I will not. Botrychococcus is the highest, but there are certain problems in growing Botrychococcus, but there are many other algae which show good amount of lipid which could be used. And these are some of the, particularly for the students, they will feel familiar with these: Botrychococcus, Chlorella, Senedesmus, Senacococcus, Chlorococcum, Nanochloropsis, Tetraselmis, Chlamydomonas, Nitschia. These are some of the potential candidates for biofuel production. Now, as I told you, lipid, one should know also lipid quality, and one has to screen large number of the strains and find out how many are saturated fatty acids, how many are monounsaturated fatty acids, how many are polyunsaturated fatty acids, and so on. Now, once this is done, then the alga is grown on a large scale. I'll show some of the pictures, and these are used for a biodiesel. Biodiesel is a mixture of monoalkyl esters of long chain fatty acids, and in case of alga, it is a renewable. And I think my previous speaker has mentioned about triglycerides, etc. I'll skip this. And then this algal oil is converted into biodiesel through process called as a transesterification, and then you get a biodiesel which could be used. See, the advantage of the biodiesel is number one, it is a pollution free, and if you add say 10% or 20%, that much of biodiesel obtained from crude oil would be saved. That is how you would be saving, number one, the costly material, particularly brought in using foreign exchange, and number two, again you are reducing the polluting agents. Now, since time is very important, I will not go to this. This is one of the example where how fatty acid profile is used to find out whether biodiesel that is produced is suitable or not, because that is something used for, since there's to know melting point, boiling point, ketone number, oil stability, density, viscosity. Because, see, as you know that the oil should be having a proper viscous, because ultimately, as you know, from the tank, it is pumped and brought to the engine. So viscosity is a very important one into this. Ketone number is something related to the ignition and ratio of polysaturated, unsaturated, density. These are important. Now, this is something, can you say this, sir, everyone? Hello? Hello?
Yes, sir. See this.
Okay. Now, this is something I should say that I'm a lucky because I'm a part, I was involved in the process. This is a biomass production and oil production facility that Reliance has established at Gagwa. The area that is covered is about 160 acres. And here, now this is the area where the laboratory setup is there, where the isolated strain is brought and upscaled, and then the upscaling is done to a larger volumes, and from this ultimately it goes to a final area where you have a ponds whose volume is about 5 hectares. So now the area that is chosen is a Gagwa near Jamnagar. The advantage is sea is very close by. So seawater, which is plenty of the available, one need not depend upon the fresh water, which is, as you know, that its availability is less. Number two, this is the area where you have only few days of the rain, and rest of the year, enough sunlight is available. But there are certain problems in this area. In summer, temperatures go, the aerial temperatures, ambient temperatures go as high as 45 to 47 degrees centigrade, where the pond water temperature goes up to 37. And during winter, the temperatures fall to 4 to 5 degrees in the ambient temperatures, and water temperatures fall up to 7 to 9 degrees centigrade. So you need to have an alga which should be able to tolerate a temperature fluctuation from 7 degrees to 37 degrees. And luckily, because we screened large number of strains starting from Ladakh to down Kanyakumari, from Rajasthan to Kolkata, entire India we covered. And luckily we got a strain. Initial strain that we got could grow for 1.5 years, but ultimately later on it failed. As I told, the resilience of algae is very important in this respect. And now we have a strain, it's called Picochlorum, which is able to survive, grow very well in a proper density, giving a proper crop. So, it can be successful one, but as previous speaker told, that cost is very important. The industry cannot depend alone on oil production. Therefore, they have to go for other products like natural products which are present, which are available all over the world from algae. Then, besides natural products, like one can also, algae can be used before oil extraction. Those can be used for a pigment extraction. Pigment can be taken out and then alga can be processed. Before even oil extraction, protein cannot be isolated, can be separated and used as a source of protein. Now, pigment is one of the very important one, and our students know that pigment, in case of algae, is used for a classification: green algae, brown algae, red algae. Now, luckily, these pigments are found to be commercially important one, like carotenoids, beta carotene, phycobilins. Among these. Sonali? Among these, astaxanthin is very important one. Astaxanthin is obtained from Hematococcus with the stress, say it could be a temperature or other stress, it starts accumulating astaxanthin, and astaxanthin has a huge market. Then here is the alga. So one can use for astaxanthin production as well as for oil production. Here is a Dunaliella which grows in a salt pans, which produces beta carotene at higher salinities and higher temperatures. Now these pigments are used as a natural food colorants in cosmetics. Although synthetic coloring reagents are available, but those are toxic one, and therefore nowadays people are giving preference to the natural colorants. Now these are some of the products that are available in the market. Now the next pigment that is used worldwide is a phycocyanin. This is from spirulina, phycocyanin. This is a phycoerythrin obtained from porphyridium. Porphyridium is available in India, is present in our country. It's grown on a large scale using tubular photobioreactors, and this is a phycoerythrin. Now, if we look into the applications of pigments, those are in food industry, those are in pharmaceuticals, those are in cosmetic industries, those have a therapeutic medicinal applications also, nutraceuticals also. Now, the other byproduct nowadays is very important is what is called as polyunsaturated fatty acid. It is said that what you eat is very important one, and the diet with omega-3 and omega-6 fatty acids is very much helpful, and particularly these are used in infant formulations. The development of brain particularly is depending upon whether enough amount of PUFA is present or absent into this. I think this is what I thought I should convey to the students and others. Now, before I say thank you, I would like to thank the organizers, Professor Kumar, head of the botany department, K.K. Sau, Professor B.N. Verma, because of whom I am now in front of you, other authorities of the university, students, and others. Thank you for patience hearing.
Okay, thank you. Okay, thank you, Professor.
Welcome, sir. If you have any queries, if anybody would like to ask, I'll be happy.
Sir,
yes.
do you think that in coming days, the production of biofuel will be the supplements and the main resources?
Yes, sir. The previous speaker talked about genetical modification, all those things. Certainly, yes, that is another aspect. But my own experience has proved that even native strains are equally important one. And now with that, now the Reliance is now working on protein from the same strain, as well as they are working on a production. See, once you separate protein, then the biomass used for oil extraction using hydrothermal liquefaction, and rest of the biomass is used. They are planning to use for as a field for aquaculture, field for fish, field for shrimp. So if you get a more, see, there are two trends. One, cost of the crude oil is going on. It's increasing day by day. See, when I was a student, it was 1 rupees per liter. Now it has reached 100 rupees. The diesel has reached now 80 rupees. And plus, with the value-added substances, certainly it will be in reality.
Okay, sir. Great, sir. Thank you very much, sir.
Sir, what is the cost effectiveness in coming years as compared to the natural?
Currently, it is highly costly. It is costly.
One question, sir. Here. Biofuel is dangerous for human beings, yes or not? One question.
Hello? So the question is,
Biofuel is dangerous for human beings, yes or not?
sir, one of the student is asking whether the biofuel is dangerous or not.
It's not dangerous. It is safe, very safe.
Thank you, sir.
Oh, 1 minute, sir. 1 minute. 1 minute. Let me interfere. Biofuel obtained from algae is safe, but biofuel obtained from jatropha is not, because during extraction of jatropha, there's a toxic substance that has been detected is a jatrophicin, which is highly toxic.
Thank you, sir.
Okay, sir, thank you, Professor Chabale, for your nice and elaborative and informative lecture.
Thank you, sir. Thank you.
Thank you, sir. Okay, sir.
Okay. Done.
So, hello. So right from early in the morning, from 10 a.m. up to now, we had the lecture of different resource persons, Professor Chowle, Professor Samit Ray, and Professor J.B. Casey. In addition to the resource person, altogether 5 research scholars and the teachers of the Mitra University have read their paper. So now it is a very brief session, and Professor K.K. Sahu will summarize everything, and with thanks to all the participants. Okay, Professor K.K. Sahu.
Thank you very much, sir. Today, we started the session at sharp 10 o'clock here in Muzaffarpur. It was a very chill day, and surprisingly, physically, almost 100 plus were present in the hall. The inaugural session was presided over by our Vice Chancellor, Professor S.P. Singh, and co-chaired by Professor Dolly Sinha, the Pro-Vice Chancellors of this university. Professor Ratan Kumar Chaudhary, the Dean Faculty of Science, the inaugural session. He, John 3 digitally told about the different views regarding this seminar, and all are immense economical aspects in research and its other aspects were discussed. In the first technical session, Professor H. in his very marvelous speech, gave a lecture on the algae inflammation. He showed different aspects of the utilization of the pigment in a different ways, in the cosmetics, in the cake, and different types of uses. And they showed a different line, different usage of the different ways of our human technology. In the first session, it was a paper presentation by scholars and students. All together, 5 research papers were read by the students of this university. However, altogether 32 papers were received for the presentation. And far the time is concerned, and only research scholars presented their paper on different aspects of the well. In the post-lunch session, Samit Ray from this party, Shanti Niketan, gave the detailed aspects of the algae and biofuel. He gave the broad idea and the different constraints that have been used in the productions of the biofuel. And last speaker, Professor B.B. Chocolate from the Pune, he gave a marvelous dimensional different aspects of the productions of biofuel, and he also compared how the biofuel of the algae is much great use and beneficiary in compare with the other biofuel productions, and even comparing the lipid productions, and he showed that there is a less area, less land, less environment we can do in compare with the other land habiting plants. We also showed that the in coming days, naturally biofuel will be the one of the main resource of the fuel to be used for human beings. During question answer, it was came to know that the biofuel produced by the algae is a non-hazardous, while the biofuel produced by the other plant is a dangerous or hazardous for the human being. It was also a very important thing noticed by for the audience. So, in this day-long seminar with the 3 stalwart ecologists gave their marvelous speech to us, and it is a much benefited for our students and research scholars. Thank you all the participants around India who has participated with us in a different way. Once again, thank you.
The seminar is over. Thank you for all the participants.
Thank you.
Thank you.